will be not very tired and will be full of energy to also try fixing with gene editing the genetic polymorphisms they don't like. I will explain how actually you can design a CRISPR guide for a gene and how you can put it into some vector like a viral vector and even get a quotation, so it will be an exercise. Yeah, and then let's first tell me my story, how actually this project started. So as I mentioned, I am in the same systems biology of aging group lab as Larissa. Also, I am involved in Cell Fabric. It's a startup created by part of our lab. And also, even before joining the lab and creating a company, I have always been active in different longevity and transhumanism organizations, because I believe that we should do something about solving aging. And I also an active open source developer. And when I joined the systems biology of aging group, I actually thought that, okay, I know a little bit of bioinformatics, and I felt a bit guilty that I don't use it in an everyday life. You know, it's like being a shoemaker, but not having any shoes on you. And this problem puzzled me a lot. And eventually I decided to sort it out. So first step I did was to sequence myself. And as probably at least 27% of you know, it's very cheap now. So probably the cheapest solutions are Nebula and Dante. You can get 30x coverage, full genomic sequencing with Illumina for around $300 to $500. Many people heard about other services. Probably everybody heard about 23andMe, but it's not a proper full genome sequencing. It's just sequencing some spots in the genome, like 650,000 variants. If you don't have those variants, you don't. So you can only see the variants that they have in a chip. And approximately when you sequence with proper like full genomic sequencing, you get at least 4 million variants. So you got way less than you typically get from sequencing. And there are other also services like Ancestry and Self-Decode that actually, in my opinion, now provides one of the best health and genetic recommendation. The second thing that I did, because I was doing a lot of bioinformatic pipelines, okay, I got my sequencing, and let's get to the very raw data. Let's apply the latest tools. And I started publishing this code to GitHub in an open source way. And at some point, I discovered that one anonymous guy with a new nickname Newton Winter with a rabbit on the user picture started to contributing to my code. And from this point, actually, it turned from just a personal project into something something more serious. And now I give a word to Newton Winter. So please follow the white rabbit.
Hello, my name is Newton Winter, and I would like to share history. At the moment, I found DNA-seq repository. I already had my genome sequenced, I got a whole genome sequence. And I had raw reads, but I was not satisfied with reports the commercial service provided because these were not transparent. They didn't share how did they came to conclusions they provided based on my genetic data. And I also had raw reads on my hands, so it had a potential for usability. So I decided to use a open source pipeline I found, and that's how I joined the project because I started using and contributing right away. So the sequencing pipeline consists of assembling raw reads against a reference sequence. Commercial services that are widely available provide reads with average length like 100 to 150 bases. It's so-called short reads, and after these are aligned to a reference sequence, variant discovery is done, and those variants are then analyzed and turned into human-readable reports and conclusions. So assembling a genome from scratch is a really difficult task, and it's not only computationally hard, it requires a lot of data. Thankfully, this was done in Human Genome Project that done de novo assembly of human genome. And thanks to that project, we have a reference sequence. By the way, commercial services usually use outdated versions, like previous builds of human genome, and this was exactly my case here. So when you have your raw reads, like a shredded piece of papers, you can align them to a reference sequence of human genome that is also openly provided and available for downloading and usage. And this is done by commonly known Burroughs-Wheeler aligner algorithm. DNA-seq pipeline chose to somewhat shrink amount of job done within the pipeline because technologies develop rapidly, and there is a rather new variant calling algorithm called dict variant that was introduced into the pipeline, and after comparison with other algorithms, it turned out to be the best. It is algorithm that is done by Google. It's based on deep learning, and it's a really clever idea, I'd say. Neural networks are really good at analyzing images nowadays. So what they did, they basically made a representation of short reads alignments as images. And the convolutional neural network showed its excellence. This algorithm showed like 99.9587% accuracy, absolutely acing the test. And it also excels in solving difficult cases of variant calling when algorithm needs to decide whether the variant exists, or it's a reader, or it's a heterozygous variant, etc., etc. So when your genetic data is assembled into genome and variants are called, there are numerous of types of changes that can differentiate your genome from a reference genome. Changes can happen anywhere. Mutations can happen anywhere in coding regions, in non-coding regions, in promoter regions. So when a change of the base results in change of genetic sequence, but the protein sequence that's produced upon it does not change due to genetic code redundancy. Such changes are called silent. Mutation that results in amino acid substitution is called a missense mutation. There is also a nonsense mutation kind when the codon for amino acid is replaced with stop codon, so the protein turns out truncated, and this usually leads to some kind of deficiency or this protein doesn't function properly. There is also a more severe change, so-called frame shift mutation. It is when genetic code is either inserted or deleted. When it happens within a coding region, it results in a severe change because the resulting protein will turn out entirely different from what it was initially. And this is usually harmful, but not always the case. Different is a very sophisticated algorithm, and making it was only possible because of large amounts of data collected from sequencing. These algorithms different Bureau's-Wheeler alignment variation we use, namely Bureau-Wheeler's aligner MEM2, which is somewhat hungry for memory, but it works twice faster. These are containerized. Docker containers are used. That allows for easy upgrade, namely different got to version 1.3, and it improved it also already perfect accuracy even further. So as BVA MEM2, it's also somewhat improved, and using containers allowed us to quickly and painlessly updating these parts of the pipeline without need for keeping the environment clean. All this is available on GitHub. You can look at it for yourself, and here's a link. And I believe now I give my microphone back to Anton. Thank you.
Yeah, so with help of Newton Winter, we reasonably quickly got bioinformatic pipelines done. So we got from raw FASTQ with the latest reference genome, not old one like in Dante, to VCF produced by Deep Variant. And the next steps was actually annotating the variants because you have to get meaning out of which mutation. Originally, we used web that some of you may know from Ensemble project, variant effect predictor. Then we switched to OpenCravat, which is much more convenient to use because it has an app store and many databases are represented as applications there. The problem is that when you start working with this, you see like huge number of different annotators, and we didn't know much by the time what to do with them. So of course, the most obvious solution, take all of them. And we selected all annotators. And we start with the second problem, is that after you annotated everything, you imagine you have like 4 million SNPs, and well, maybe several thousands. Actually, here it's 4,700,000 SNPs, and you get tens of thousands of annotations, and you don't really have time to read all of this. And at this point, we actually got stuck a little bit, and we were continuing in background mode until Gitcoin grad round happened. So a friend of mine, Lawrence Ion, told me that there is an opportunity for longevity open source projects to get some cryptocurrency funding. And I was thinking what to apply for, and it was extremely short notice. So I just applied within I think 1 or 2 days. So I remembered that I had an idea to actually do longevity annotator, because what I really lacked in self-decode and in Dante reports is information about variants associated with living longer. And also, I somewhat lacked more transparent and more serious polygenic reporting. So it was just within just 1 day, we had to take a logo and make a name of the project. And I made very original name, just DNA-seq. And also as a logo, I took 3D structure of DNA from PyMol. With Newton Winter, we expected originally that maybe we can get $1,000 or $2 so we can write a plugin. We also added Olga Borisova to the team because we needed really experienced medical geneticist, and we were really amazed that actually with matching, so the way how Gitcoin works is that it's like crowdfunding in crypto, but all funding you get is matched with so-called quadratic formula from the sponsors, and you get much more matching that you get funding, and they try to facilitate small donations. You get higher matching for small donations. And we got like $70,000, and we thought, okay, guys, maybe we should get really serious. So we made a budget for 1.5 years, and we increased the number of features we wanted to do substantially. And after we got this funding, I think it was December last year when the campaign happened, and the end of January when we got it, we actually started thinking, okay, what we should first do. And surprisingly, it was actually something that any normal people do in the very beginning. Let's first see what is possible, and what is heritable, and what is the most interesting information. Because before that, we just went, okay, we see the data, let's do pipelines, let's do variant calling, and let's see where it will get us. So here, in terms of what is possible, so here you see a picture from one of the genomics paper, and you see that some diseases like type 2 diabetes, like gestational diabetes, are highly heritable. You get like 60% chance to get them based on your genetics. We also looked into some other traits that are not necessarily health related. For example, what is the chance of having a higher IQ, or what is the chance that you will be homosexual or bisexual, and autism chances, depression chances. So here you see a Nature paper where they give two parameters. So in terms of heritability, you have to understand that you can check heritability based on studies of twins and families. So because twins have the same genetics, you can have some ideas how many of the twins have the same phenotype in terms of diseases or IQ. And with families, it is less certain, but also you have way more data on families because amount of twins is limited. And what is interesting, you can notice here that the blue is much higher than red, and red is predicted heritability by GWAS models. I mean, technically from your genetic polymorphisms. So the reason why some traits are higher in families and twins partially is because we share not only the same genetics between twins, but also similar surrounding. But the other reason is actually performance of many genetic models is quite poor. And that means that if we will have more data and if we improve our predictive models, we can actually get accuracy similar to what is in blue. Then we looked into longevity. So regarding longevity, we all know that if we compare species, we see huge differences. Like here you see a picture of the same turtle, and you see that this turtle outlived Darwin, outlived many other people. And if you look into comparative genomics and transcriptomics, you can actually see that, for example, among mammals, it might be possible to live like 200, and according latest estimations, some bowhead are suspected to be like 260 years old. And for vertebrates, probably 400 or 500 is possible. In the same time, when we compare within species, things can be quite different. And a lot also depends on the way how species live. So here on the right, you see survival curves. So usually survival curves, you start with some number, for example, 100% or some number of people like 1,000. And during lifetime, some of them die. And you can see that for humans, most of the people die relatively late. So like close to 80, 70, 90. So we have this shape. And partially it's because we have good medicine. While for short-living animals, usually they're just eaten during their lifetime. So the difference is much serious. And if you look into the most interesting categories that we have, centenarians, you can see that actually there is much more centenarians than most of us think. So it's like 500,000 of people who are older than 100 years old. That means that even though it's pretty rare, guys, there is still a lot of data to think about. And there were several papers about heritability of longevity. Unfortunately, if you look into like general heritability, you actually see that it's pretty low, like 22%, 18%. And for us, it was very disappointing. In the same time, we don't only like target just probability to live to some age, but we also look into risks of diseases that can seriously shorten our lifespan. And also, it's not really actually simple, because in some research, like here, you can see that the probability to live until like more than 90 or 100 is more heritable than just probability to live longer. And that means that exceptional longevity has higher hereditary components than just regular longevity. So technically, how you live, how long you live, for example, 1980, maybe 85 is more defined by your lifestyle, while your chances to live more than 85 or 90, they all depend significantly also if you have some centenarian genes. So for extreme longevity, it's a bit more heritable. And of course, we should learn why, and because maybe we can do some gene therapies to fix the issues for ourselves, I don't know. And what is also interesting, it's also exactly not very simple, because the amount matters. So it's quite a recent paper here. So you see, what you see is Huntington disease. It's actually quite unpleasant neurodegenerative disorder. And what is interesting, so it's based on TAG repeats. So you have some amino acid repeats within the genes. And if you have too many repeats, then you get this unpleasant disease. You see a picture with old ladies that is probably not really happy. And the thing is, but if you had the medium number repeats, more than for wild type people, but less than for Huntington disease people, you actually have higher chances to become centenarian. So it's like something can, in some dosage can be a poison, but in some dosage can be a cure. Knowing this, we actually started investigating existing longevity databases, because we are also interested not only in our genome polymorphism, but maybe we can find some ideas how we can, some gene editing ideas. And regarding aging, so there are several important databases on aging. So some databases are about different species. So these are databases that I recommend for you to see if you are curious about extension of lifespan on model organisms. So it's GeneAge by João Pedro and also by Robbie Takuto, our PI, who is was first author in that paper. And the other one is about interactions between different genes, because genes often work together. And if you have synergy between genes, you can get way higher life extension. And this database GeneAge, actually we probably have its first author, Gabriela Buno, in the chat. So if you're curious, you can ask her. And for we can also see that from those databases, we see that for animals, there are pretty good results, like lifespan of mice can be extended 50%, and in some cases up to 100. For worms, it's like 10-folds. And in terms of what we know about longevity about humans, are there any genes that we can modify, like centenarian genes, to live longer? Unfortunately, there is less information, and for only some of the genes, we are super certain that those polymorphisms are really extending lifespan. And this is what you see is a longevity map database by Joao Pedro group. And there you can see what we know so far about pro-longevity polymorphisms. And when we started writing our longevity annotator, our first step was actually making a module for OpenCravat, the annotation platform we are using, to actually check which of those polymorphisms we have in our genomes. And also during this work, we also made many extensions of the data in the database. And with this, I am giving a word to my colleague, our chief medical geneticist, Olga Borisova, and she will show our first pre-alpha example of longevity module.
Yeah, hello. Thank you for such a nice introduction. Indeed, I'm Olga Borisova. I have been working in St. Paul's German Diagnostic Center in Odessa previously. We worked with human genetics, with longevity variants a little bit, mainly with diseases. And also I have my own project related to mitochondria in aging. I enrolled project Just DNA-seq, I think, in January, and really happy with it. So we have longevity databases in several, where some important, according to multiple studies, genes are listed, and it's valuable information. But we want to know, what about us? What about me? Am I a carrier of longevity SNPs? Yeah. So based on this database, Pedro database and OpenCravat platform, we will tell about it a lot of information a little bit later. We created an annotator. I just sent you a link to it, so you can see it in the chat, and you can just link it and do something with your hands for the longevity variants. So let's look what we have now. It's a lot of stuff still we have to do with it. It's just a hello world version, but you can see the variants which are associated with longevity according to the database we used. We still have some genes here listed, which population was studied for this association. And if you just click, you can see the study design, which study studies this polymorphism and which result they gained. And also you can see a cDNA change, not just RSID, which reference and which alternative base happened. Okay, so if you are familiar with longevity topic, you can see a lot of well-known genes here, like ApoE, FOXO3, TP53, etc. And we also want to do, and which is the most important thing, is not just provide information whether you are most likely to be a centenarian or not, but also give an available data if you are not lucky, that happens with some gen variants. What does it mean? How can you affect them? How can you prevent their negative impact? And for that, we will give some additional info into our annotator. Okay, let's, for example, talk about some genes. Now there will be a piece of theoretical biological stuff. I will try to make it quite easy to understand, so without some difficult words for those who are not biologists. Let's find UCP genes. You can use just Ctrl F or just scrolling UCP genes. You can see that it's an annotation which was made from Anton's genome. We're always using it like an experimental model because he has his genome already sequenced and he allows us to play with it as we want. So what are UCP genes? We will talk about it now. And let's briefly So you will see it's not just a line in the annotator, it's a whole story inside you, inside your cells. And none of us is a superhuman. Yes, but unless we act towards our well-being and longevity. Okay, I will just change into the just one second. Yep, okay. So you can see from the annotator, the link I have sent you, or just following the slides, that there are several UCP genes. Actually, there are 3 of them. They have high homologicity. And Anton is a lucky one who carries one allele which is associated with longevity. So you can see it in the conclusion from our annotator. But what does that mean? So if you are not so lucky and don't have this favorable allele, what should you do? So what's UCP are? They are genes which are coding proteins related to mitochondria function. I am totally in love with mitochondria, so I will tell about it a little bit more. And I think it's one of the most important things in the longevity at all. I will briefly remind for those who are not biologists. As you all remember, mitochondria are intracellular organelle and almost, okay, what happened? Okay, hope you see it well. I don't know why it paused. And mitochondria main function is generating energy in ATP form. It's not the only one function. They have a lot of like immunology, sterile inflammation, etc., etc. But all of us know from our school programs that mitochondria is a powerhouse of the cell. It generates the energy in ATP form, which cell can use for biochemical and other processes, to fold proteins, to digest something, etc. And ATP is a unique molecule which is an energy currency for all of life species, actually. Okay, how is energy generated? Just briefly remind about it. The food we eat, and it really doesn't matter whether it was a tasty burger or kartoffel-free, I don't know, French fries, or healthy avocado and salad. All this food is going into it's digested, making some simple molecules, and the simplest one is going to the mitochondria. It's calling acetyl-CoA. It goes to the Krebs cycle, and after that, we have our energy. This is the simplest atom of hydrogen. It's all we are gaining our energy from. It dissipates into the electron and proton, and electron going to the electron transport chain, which is formed with several protein complexes. So it's like current in the electric wires. And during electron migrates from one complex to another, some small amount of energy is generated. And this energy is spent to push down the protons of hydrogen into the intramembrane space. So now mitochondria, they look like an accumulator. They accumulate a lot of energy, like proton motion force. And also into the inner membrane of mitochondria, ATP synthase complex is located. It's like a nanovehicle. And when protons goes back through ATP synthase into the mitochondria, ATP is generated. Okay, it looks like water in the water tower. So when you're pulling water and then pulling them down, it can make some work. It's the same for mitochondria. But overcharging the membrane is not always good. So if you don't need so many ATP, but still continue eating here, still continue supplying hydrogen into the mitochondria, it may lead, so this electron leak from the electron transport chain and give start to reactive oxygen species, ROS molecules, which can damage macromolecules, lipids, proteins, DNA, RNA, etc. And it can lead to the oxidative stress. So overcharging is bad. And nature has created the mechanism which protects mitochondria from this stuff. It is called uncoupling. A cell is doing it, mitochondria is doing it with special proteins which are called UCP. Remember, we talked about UCP genes. UCP proteins are located also in the membrane of mitochondria, and when the membrane is overcharged, it just let protons come into mitochondria without ATP generation. It's fast, it's simple, and the energy is just dissipated like a heat. So it was a simple process which can prevent overcharging of the membrane. It's a biological uncoupling process. This process is calling uncoupling. So it uncouples ATP generation from electron transport change. And also it can be like chemical uncoupling. Some chemical compounds can cause uncoupling as well. Okay. So one of the most important, best known examples of uncoupling is brown fat. You know, a lot of UCP proteins contain brown fat mitochondria. This is a good fat. Brown fat is a good fat. And the bears have such fat. All the fat can be burned fast and easily without any metabolic syndrome. And the formed heat allows them to survive sleeping during the winter. And some of our white cells can probably also go brown. And there is a lot of approaches. So it's like a dream of humanity. So have a fat that burns themselves without any damage. And there are really a lot of approaches in longevity-related fields, sleeping in cold, taking capsaicin and other hot compounds, taking fats, taking some medicaments like actually tadalafil cause fat browning. It's like Viagra. It's an interesting adverse effect of such a medicine. And also some interesting experimental approaches like nanocarriers with gene therapy modules, which inserted into the white adipocytes and make them brown, etc. But uncoupling is needed not just for fat burning. Mild uncoupling is associated with exceptional longevity of bats and mole rats. You know these animals, if you are interested in longevity. They are small and have like high metabolic rate, and their prediction its lifespan is not that high, but compared to regular mouse, which lives like 2, sometimes 3 years, a naked mole rat can live like about 30 years. So they developed a mechanism preventing mitochondrial membrane depolarization, and it is called like mild uncoupling. In mice, this uncoupling mechanism began to decrease significantly after the first year of their life and almost completely disappeared by the age of 2 years. And in the tissues of naked mole rats and bats, on the contrary, the process of moderate depolarization was active and is active for most of their lives. And this is one of the mechanisms why do they live so long. And another interesting stuff is uncoupling proteins is, you know, the term that our brain boils. Actually, uncoupling proteins, they exist not just in fat. UCP1 is mostly abundant in fat. UCP2 is the most abundant uncoupling proteins in our tissues, a lot of them, and brain tissue as well, neuron cells as well. And in the neuron cell, uncoupling forms up our brain. You know that synaptic transmission, it's kind of a chemical reaction of releasing neuromediators. And UCP working gives them move faster and release faster, and so it helps synaptic transmission to become faster. And indeed, problems with UCPs are associated with cognitive impairment, like not just dementia, but memory impairment, attention impairment, etc. So it leads to the developing of uncoupling to survive theory. The genetic variation of human UCP genes is associated with longevity, and that is what we have shown in our YES database. That there are several UCP genes which were found in Anton's genome, and they are positively associated with longevity. But what to do if we are not that lucky that we don't have positive genetic changes in UCP genes, for example? Actually, a lot of biotech companies are working on developing mild encapsulators, and they are potentially powerful drugs for treatment of age-related diseases like metabolic disorders, metabolic syndrome, diabetes, etc., and neurological disorders, neurodegeneration. And some mitochondrial startups are working to create safer ones. So they work just in a really narrow range of pH, and if they uncoupling for some time and then pH changed and they just stopped working. So it's quite safe, and they causing really mild uncoupling, not leading to ATP generation decreasing too much. And I here just put some links for those who are interested in just some biological startups, etc. You can look at this. Continuum Bioscience and Mitochondrial Pharmaceuticals. They have a little bit different approach, and they are focused on different stuff. Like first one is focused on metabolic disorders, and another one on neurological disorders. So if your UCPs are not working well, mild encapsulars may be a good choice in a later life, especially. And a lot of famous longevity drugs are mild encapsulars. All of us know aspirin, metformin, and they act partly through uncoupling mechanisms. Okay, so it was like a little biological theory. In our second part of my talk, we will go to some practical stuff. And now it's my pleasure, a really great pleasure, to introduce our collaborator, who will tell you about OpenCravat and OAKVAR, which are software, which is open source software using which you can analyze your genome, and we will do it. So let me give a talk to our next speaker, Rick.
Thank you. Hello, everybody. Okay, I'll share my screen. Just right. My name is Liang-Guk. English name is Rick. I am from the company Oak Bioinformatics, and I'm going to introduce OpenCravat and OAKVAR here. So your DNA and DNA sequencer, and you sequence your DNA, and then you get the file of your variants, and then we will use some software to get to the meaning of our DNA sequences. So then this software, so OAKVAR and OpenCravat, is one of those software programs to get some meanings to help interpreting your variants to know what your DNA sequences mean. And the history of OpenCravat and OAKVAR started in 2012. So I was the main developer of Cravat web server. It was a Johns Hopkins project. And that time it was just a web server with about 15 annotation sources, such as CleanVar and Cosmic. And it was for cancer variants only. So it analyzed cancer variants and provided annotations. Then 2018, the project OpenCravat started by the same lab, and I was the lead architect of that project. Actually, I proposed making a general and also locally available Python package to annotate the genomic variants. And that resulted in OpenCorvet and as a Python package. So you can locally install and use your laptop or desktop to analyze your own genome or other people's. And also the web server is still available, run.opencorvet.org, which is used in this workshop. Then this year, I launched OakVar. It's further developed version of OpenCorvet, and it's backward compatible with OpenCorvet. And still, it's a Python package. And this is the GitHub. And there's no public server yet for OakVar. So if you want to just conveniently go to a web server and upload your genome and get the annotation data, then run.opencorvet.org is the place to go. Now, what does it do? So I will just use OakVar for this presentation. So Nebula Genomics, and 23andMe, and other the sources that Anton described will provide these files, basically VCF format files or 23andMe files, etc. Then we will run OpenCorvet or OakVar to get annotation. So what does it mean, annotation? I will explain in the next slides. Then we will have a collection of annotated variants. So then it's stored as a database. So that's the part of annotation. It basically means we add a bunch of additional information to each variant you obtained through these companies' sequencing services. Then the next step that we need is interpretation and getting the meaning out of those annotated variants. And to help us know the meanings of our variants, OakVar will generate, and OpenCorvet will generate the reports, and there are web apps, and it's connected to, actually it's OakVar, Python and R scripts. So these help getting the meaning out of these annotated variants. So for example, this is one VCF file, the portion of it, and then this is what we get when we get the sequencing data from a company like a Nebula Genomics. Then we have chromosome and position, and then the alleles, and that's it. And then some statistics and some annotation. It's very difficult to know what it means. So we run a program like OakVar to get the annotated variants. And then this is one example, web browser-based, the result viewer of OakVar with some graphs and statistics and other. So it just helps us knowing the meaning of these annotated results. So what is annotation? There are a lot of databases out there like CleanVar, Cosmic, CAD, Revel. They have their own database that adds the meanings to these variants, but they handle different aspects of each variant. For example, there is an allele frequency database like NOMAD or 1000 genome, which means how frequent your variant is. And CleanVar will tell you if your variant was found in any pathological actual clinical cases or not. So these different annotation databases will tackle your variant from different angles. So the idea of OakVar and other tools also is to combine all these different sources into one table format, so that we can see all the different aspects of each variant So variants are in different rows. So they are annotated variants. And in the OAK-var is a Python package, and it's a modular. The architecture is modular. That means each annotation source can be packaged into one Python module, which can be imported by OAK-var. So it's very easy to make your own custom module, which is done by this longevity group also. So what you need is just a data source and a Python script that will use this data source to produce that annotation output for each variant. And the OAK-var will handle combining all these different annotations into one database. So annotation variants and OAK-var and annotated variants using these different data annotation sources is Python modules. Then usually we need filtering because we have millions of variants for each person, and we need some filters to bring those number of variants down to some manageable number. And then using those filtered variants, then we generate reports, apps, and further analysis to get to the meaning of our variants. So for example, let's say this is one variant you got from the sequencing company, and using the CleanVar and OLE frequency module, the OAK-var annotates that from the CleanVar, this variant is pathogenic from some clinical study, and OLE frequency was 0.000. That means it was not observed in one case, at least out of 1,000 people. So it's very rare. So that means this is a very rare and pathogenic variant. So that, of course, you will have a lot more variants than just one. So we need, when we use filter, give me only rare and pathogenic variants. Then the second one will be filtered out because the clinical implication of that variant was uncertain. It was now observed definitely as pathogenic. So you will have just a first variant only. So that's how we get to some meaningful collection of variants using the filter that we set. And then I'll get to the meaning part shortly. Now, recently, well, I think it was the last week, this company called Ultima Genomics, they announced that they are targeting a $100 genome, which means you'll be able to sequence your genome with at the cost of $100. So it's quite big news because currently Anton showed the slide showing the price of $300 or $500, etc. So it used to be a lot more expensive, but still it's not that cheap. But with $100, it's I think it's really affordable. Then almost it's genomic sequences available to the mass. Then it's democratization of genomic sequences to the mass. Then what OAK-var is aiming is the democratization of genomic analysis. So we will have variants like let's say everybody can have their his or her genome sequence. Then ideally, I think we should be able to analyze our own genome with our laptop. And people have different skill sets and different preferred ways of analyzing the data. So some graphical user interface-based approach, or some report, or some people are just familiar with Python or R. So with OAK-var, you can connect to these different modalities to go down that the road of finding meanings of your own variants. So for example, since it's Python based, you can use Python Jupyter Notebook or just Python script to connect to OAK-VAR annotated variant database. So you can query your annotated variants using the Python. Also, there is an R binding to OAK-VAR. It's called the ROG-VAR. So you can import or just run the OAK-VAR analysis, and almost anything OAK-VAR can do, you can do inside R also using this R binding library. And of course, there is a web browser based result viewer. So you can filter and just examine the data. Also, it's possible to make a custom web app. So the concept of web app is OAK-VAR is the platform that powers the web-based applications that access the annotated data variants. So this is one example. This web app was developed for molecular tumor board group at Johns Hopkins University. So it was their internal web app, and you can see it has some nice formatting and custom stuff here, but it was powered by OpenCravat. It was made before OAK-VAR. And finally, because it's a Python based, it can send the annotated variants as a pandas data frame using also the filters if you want. Then, because it's a pandas data frame, you can feed that to AI or ML libraries, Python-based libraries. So this OAK-VAR can be the gateway to use your annotated variants for AI or ML-based studies. So the OAK-VAR, we aim that to be a genomic analysis platform for the mass. So oakvar.com will lead to the GitHub repo, and you can just install with pip, PyPy. And if you have any questions after this workshop or anytime, then just please send me an email. Okay, so that's all from me. And
okay, thank you very much for such a clear explanation. I'm sure everyone will be just exciting now to analyze something personally using OpenCravat or OAK-VAR. Thank you very much.
Thank you.
So we will back to use questions, I hope, after closer to the end of our seminar.
Yep.
Yeah, okay. So now it will be again me talking. And I'm very excited to be responsible for some kind of a technical stuff. Because actually I'm a disaster person with all of related with technical stuff. We have listened to a perfect talk about OpenCravat, what it is, OAK-VAR, like a variant of it. And okay, to make things simple, let's do a small hello world example and starting analyzing and learn how to analyze your own genome, your own VCF file using OpenCravat or OAK-VAR. Let's begin with some simple example from Anton's genome. And it will be related to pharmacogenetics. I have already sent you a link to OpenCravat server. You can just open it, and until while I am talking, you can try to log in, and I hope everything goes well with it. You can use antonkulaga@gmail.com as a login and a password RCBI 2022. So if something will goes wrong with it, we have some another variant how we can work together. But please, first of all, try this one, and I hope everything will go well with it. Okay, so while you're trying to do that, I will tell a little bit about pharmacogenetics. Pharmacogenetics actually is an important thing in the personal medicine, and it is related to choosing the appropriate drug for you, the doses and forms of some drugs, and also to estimate your personal risk related to taking this appropriate medicine. Your genes are coding the enzymes which participate in drug metabolism. So you can be a fast metabolizer, like an intermediate one, or a slow one. And all of these affect the dosage of the prescription you have to take for this medicine, and also the adverse effects. So what you see in the prescription list in the instruction to the drug is usually like an intermediate variant. You can need it a higher dose or a lower dose of it. And so we will look for it. Okay, I will just, how can I just look into the chat whether everything is fine? Got it. Beginners ourselves. We will firstly work with Anton's genome and with several clinical cases which are already uploaded. And after that, you can switch to work with your own genome. Okay. Okay, so hope everyone has already logged in. Yeah. Is everything fine? Is someone having problems? Okay. I see no one telling that have problems, so probably it's just fine. Okay, so I'll just log in myself and then we continue. So if you have done it successfully, you can see something like that. There are several genomes we have already uploaded for you. So in that case, it would be faster. One of them is Anton's genome. Some of them are for our clinical cases, and some of them to show you. How actually, I will briefly tell how actually you can upload your own genome. If you haven't done it already, you will be able to do it after our workshop, just taking your cup of tea and working with Open Cravat. So first of all, you have to choose the assembled version of your genome, and that is important. Usually the company that gives you your raw data, you can find this information there, which assembled version of your genome was used. If you will choose the wrong one, you may gain wrong results. Also, there are some converters. So if you have 37 version of your genome, and you want to work like with 38, you may convert it. Then you may choose add input file. With this link, you can just choose your VCF file from your computer. Okay, and later we will talk about different annotations. Okay, now we will just look to the Anton's genome. You can find Anton Kulaga VCF file. Here are some annotators which we choose are listed. And we are just choosing the open the results here. I suppose if a lot of people have downloaded, it may take some time to download the data. And if it will work too slow, we will have to switch to our server. Yeah, Open Cravat cannot display. Yeah, okay, I will start. Finally, it's good. Open Cravat viewer cannot display more than 100,000 variants. Use the filter tab to load at most 100,000 variants. That indeed is a problem, but we can solve it using some filters. Depending on the task we want to solve, we can use different filters, whether we want to find some pathogenic variants, some pharmacogenetic related variants, etc. And we can do it using the filter tabs. So there are several lines with filters. First is genes. Samples, it's used when you are working with oncological tissues, etc. It's not for personal genomics stuff, it's more for doctors. Gen list. If you're interested to find some variants in genes and you know these genes you want to look at, you can just type a list of genes or just put a list of genes here, and you will be annotated with these genes. There are some filters which are automatical, and you can use them as well, but usually we use Query Builder. It's a kind of a filter that you can choose like by yourself what you want to do. So you see here is a little plus, and actually the filters you can apply will depend which annotators you have chosen previously when you're uploading your genome. I will tell about it a little bit later. So now our task to make a short, this window, okay, a short hello world example, and we are going to find some genes which will affect the drug response related to pharmacogenomics. We have PharmGKB filter, and there is a PharmGKB annotator, which is related to this field. And when you're uploading your genome, if you're interested in finding something related to pharmacogenetics, you have to choose this one. Then you choose chemicals. It can chemicals which is already in the database of PharmGKB and has data. Okay. Then we make plus. So if we want to annotate it not using just one annotator, we can use something to this filter. Also, there is a famous and important annotator, ClinVar. All of us are working with ClinVar. And there are several categories we can choose in ClinVar. And one of them is drugs. So you can find here drug response. Here you see pathogenic, benign, etc., etc. Drug response. Oh, we can do that. Hope it's not that fast. So we are choosing ClinVar, clinical significance, one-off, and then we select just drug response. So we combine these two filters so it would be more reliable. If you combine them, here you can change and or. And that means that using that filter, we will have variants which are annotated by PharmGKB database and also exist in ClinVar database. So it makes it more reliable. And after we have chosen the filter we want to work with, we can see how many variants we will get. We can't work with more than 100,000 variants. We can do it just clicking these small arrows, and it will show how many variants we will get using that feature. 78 variants. Okay, that is fine with us. We can annotate it using 78 variants. So actually, when you use filters, you can save this filter using this thing. Actually, it's not always working, as for me, but sometimes it works. And also you can later use already chosen filters. We will do it when we will work with clinical cases. You can just upload the filter you have already saved, and it will be automatically built into your builder. Okay, so just apply filter. And then again, it's working. It's working that slowly, I think, because kind of plenty people have connected to the server. Usually it's a little bit fast, but it depends. But anyway, it's our own genome. It's not that long to know information about yourself. Okay, so now it has annotated. Then after it, you can choose. There are also two tabs, variant and gene. Usually I work with variant tab, so you can use just it. Or no, let's put this in. Okay. And it's kind of complicated for user. It looks not really user friendly, but and when I first started working with OpenCravat, it was also complicated, but then you get used. And here you can find into the upper right corner some options. View table, view detail plane, view table plus detail plane. Usually I work with a table first, and it's a table which has gene variants according to your filter applied, and genes which are thought to impact your own pharmacogenetic response. You can see the list of genes here. And actually, columns here, it depends on how many annotators you have uploaded for genome analysis. We have uploaded pretty much because we work with different stuff as Anton genome. Now we don't need all of them. We use just pharmacological ones. So we just pulling this thing until we find farm GKB annotator. If you had more time, you can use just two annotators, and in that case, the amount of these columns will be much less. So here you can see the list of chemicals which you are supposed to have a changed response to. Here you can type a chemical you want to search. Maybe you are taking something already, or you're gonna take something. We can try just statins. All of you know that statins are commonly used drugs for metabolic syndrome, for lipid metabolism, especially in later life, and also they're quite important and quite famous in longevity medicine. People take them, that stuff. So we can see here a list of drugs which are to be affected by your genotype, and you can see here simvastatin, rosuvastatin, etc. Yep. So we can choose one line and see what gene is that. For example, ApoA5 gene. So you can find here also the information which is important. VCF info, it's a little bit to the right from the farm GKB column. VCF info, you can find zygosity. You know that you have two alleles of each gene variant. So, okay, exactly. So you can have both of them changed. So if you see here homozygous, like here, it means that you have alternative base for both alleles. If you are heterozygous, that means that one allele is alternative and one is reference. So after choosing some some line of information, you can switch to view detail plane. Here you can see a lot of windows. It's widgets for annotators. We uploaded for genome analysis. You know, it depends how many you have chosen before the genome analysis. And usually, if you will choose just 2 annotators, there will be less. So we can see variant annotation, reference base is G, alternative base is A. So if you have homozygous variants, that means you are AA genotype carrier. So we also have ClinVar widget. You can go to the ClinVar link and see what information will give you ClinVar. So it says that this variant is indeed related to atorvastatin response. So, but we need to find farm-GKB widget, farm-GKB variant. Also, there is a GNOME ID widget. We will talk about it later. It shows the population frequency of this variant change total for different populations as well. So, but farm-GKB variant, Tenofovir is not wanted. Atorvastatin. Alternative AA is associated with response to atorvastatin, rosuvastatin, and simvastatin in people with dyslipidemia. So it's our variant, and you know, alternative A, and we are homozygous. Anton is homozygous, so he's AA carrier, and the response is increased here. And here you see response, increased response to simvastatin. So yeah, probably, and here you can also see whether the data is significant. Yeah, so the efficacy is high and the response is increased. So maybe you have to think about decreasing the initial dose when you come to taking statins. But okay, it's not the only variant we have here. So our task is to analyze all of them. I hope we'll be able to create an annotator which will do it more user-friendly, and you can see all the variants like in one report, and you will be able to analyze it more easy. Okay, another one, heterozygous variant of a gene or E gene. Yeah, you know, it's a gene very important for longevity. And then again, we are changing to view detail plane. Here we can see farm-GKB annotation. We have heterozygous variant, so CT variant. And then again, we see that, for example, for fluvastatin, genotype CT is associated with increased response to fluvastatin as compared to other genomes. So yes, indeed, maybe according to already 2 variants, our response is increased. So we may think about lowering the dosage if we will need taking it. Okay, view table, again, view table. And we can have a look into the another variant, ABCG2G. So again, we are moving to VCF4. You can see that you are heterozygous, and you can see farm-GKB column. And also you see here the farm-GKB ID. You can also link this farm-GKB ID and find the additional information in farm-GKB database. Also, depending on how many annotators you use, you can find additional information in all of the columns. Yeah, for example, for PubMed, if you will click PubMed, you will get into links to PubMed associated with this gene. Okay, but we're interested in farm-GKB variant. Okay, just think here. No, you do it faster. Okay, so it's loading. You can see it's quite a huge database related to pharmacogenetics, and you see everything related to your RSID, which were found in your genome in heterozygous variant. So you can see here all of the information, if there are some. For example, prescribing info. If there are existing guidelines published by some consortiums, you will find them here, and you can see them, drug label annotation, according to these clinical guidelines. For example, increased plasma level of rosuvastatin have been observed in Asians. We recommend in starting dose, like depending on population, etc., etc., etc. Genetic polymorphism unknown that may lead to an increase in rosuvastatin exposure in patient is known. A reduction in the dosage is recommended. So indeed, according already 3 genetic variants, you are responder to statins, so it will act on your lipid profile, but you most probably, you will need to decrease your dosage. And okay, you can switch to some different apps like clinical annotation, etc. And also you can download this PDF variant. So you can also see which evidence level for this polymorphism this data has, like 3 level of 4 level, etc. Okay. Okay, so evidence level means how you can be sure in this recommendation. So the highest evidence level means that, yeah, okay, a lot of people were engaged in the study and you can trust them. Okay, so let's continue. Let's continue. So it was kind of a hello world example. Now you gain some new experiences analyzing your ECF files. And now we can go then again to the Open Cravat. I hope you haven't closed it yet, and continue doing some stuff. I'll show just a few slides and then we will go back to the Open Cravat window. So if you are back to the Open Cravat server, you can see downloaded, pre-downloaded genomes. You can see annotations which we can use. They are divided into several categories. Usually we work with variants, clinical relevance, allele frequency, genes, etc. Or you can choose just show all and then choose which you need. Okay, next one. Which annotators we usually use? So when you are first into the Open Cravat and you are just starting analyzing your genome, it's kind of complicated to choose the right set of annotators for analysis. We usually use this list of annotators, and we recommend it also for you if you will analyze your own genome. You will get the presentation afterwards, and will be able to look at it. It's like Hack-Your-Genome. We use 2 sets of annotators. The first one is to see how damaging this variant is. And another one is to find out which information we can get about this substitution or SNP. So we use Hack-Your-Genome is a tool for scoring the deleteriousness of single nucleotide variants. as well as insertion deletion variants in the human genome. So it tells us how damaging this substitution is. GNOME AD gene, I have already talked about it. It's about a population frequency. PubMed, PubMed articles related to a particular gene variant. Lingen, it's also an informational resource that defines the clinical relevance of genes and variants. ClinPred, it's prediction tool to identify disease-relevant variants. ClinVar, very big annotator, often used, contains an archive of reports of the relationships among human variation and phenotypes. MetaMap, it's for mitochondrial variants. It's not that popular, but we included it into the list. NCBI gene, it's gene descriptions from NCBI. OMIM, it's a catalog of human genes and genetic disorders. You need OMIM if you will analyze some variants related to some diseases. And then again, some annotators which will show us how damaging or tolerable this substitution is. It's REAC, PROVENE, REVEAL, SIFT prediction. So actually, we try to use just one of them, but none of them contains enough information to cover all the variants we can find in the genome. So we just combine different annotators for prediction of deleteriousness of some variants. So we will now move to the clinical cases analysis. It's real on clinical cases, and you will try to find a solution for one or another phenotype related to disorders. Clinical case number 1. You can see it's spread and loaded. It's X13S adenine S1. It's a woman, 30 years old, so quite young. She has several complaints, progressive muscular dystrophy, whole genome. You can already, okay, maybe I can already also push open result view, so it will be a little bit faster. It takes some time to. It takes some time to annotate. Okay. So she has a progressive muscular dystrophy. She did whole genome sequencing in 2017, but actually no genetic cause was found, and she got the diagnosis like myopathic syndrome of unknown origin. So she asked me a question, actually, is it worth redoing the genome or exome sequencing in Germany or some other country, anywhere, to find out whether it's genetically related disease and phenotype or no, or not. Some little overview of her complaints. Clinical picture according to her words. She lived not in Odessa, so I haven't seen her even, so I just talked to her on the phone. The first symptoms of muscular fatigue appeared in childhood, so quite early, and it shows us actually that it's probably the genetical reason for this stuff. She also has frequent respiratory infections, etc. At the age of 12, the jaw began to get very tight, so it was difficult to chew from talking and chewing, and the fatigue increased and speech became slurred, and she also observed and increased difficulty in swallowing certain types of food. At the age of 20, weakness in the limbs appeared, so it was difficult to climb stairs. And previously, multiple sclerosis was suspected, then myasthenia gravis, and nothing was confirmed according to immunological testing. at the age of 23, the state of health was revealed. She observed weight loss by 7 kilos in 2 months. From this period, it became difficult to breathe. She also mentioned that she had some problems with food digestions. Mitochondrial myopathy, celiac disease were suspected, actually. That's why she asked us, because we were famous with mitochondrial diagnostic. A lot of disorders like immunological ones, gynecological, endocrinological, gastroenterological ones were excluded, and autoimmune disease were excluded as well. So now we will go to the Open Cravat again. Accepting the question. Okay. Okay, just a second. It's a kind of slow with retrieving data, but while we are trying to retrieve it, you can. Okay. Okay, so then again, we see kind of a lot of gene variants to be analyzed at one time, and again, we have to use some filters. I forgot to do one stuff. I have to do it. I will send you now a filter, so it will make everything faster. After you download this file. Okay. Okay. Here it is. I'm just sending it to you. No, I can't send it. No. Maybe I just can't share it. Okay. Then we'll do it manually. I don't know. I don't know why, but okay, I can send you a link to my disk, and you can download the okay. Here you can see. Here you can see the folder with different files. We will use them during our talk, and you can now download pathogenic JSON. It's a filter for Open Cravat. Okay. So after we go into the filter tab, we have to choose some filter. We can apply already prepared filter for it, like pathogenic JSON. Okay, so you just choose this arrow, click on it, and then choose pathogenic JSON filter. You can download it from the link I have sent you. So while you are doing that, I will tell a little bit about this filter. So why have we chosen it? We have chosen several filters which will show us the pathogenicity of the variants. It's like cardiac zone filter. Here we choose pH red and it should be higher than 10, this filter to estimate this variant as pathogenic. Here we choose SIFT filter, and here is easy. We're just choosing prediction, one-off, damaging. So it's quite easy, and you can always do it yourself. Like SIFT prediction has to be damaging when you're looking for some damaging variants. Reveal, it also shows us pathogenicity. Here we choose score and higher than 0.5. ClinVar, clinical significance, one-off, pathogenic. ClinPred score higher than 0.5. And we can combine filters, like when we want to choose or to end, we can group them. So we have grouped filters for pathogenicity estimation, and then filters for getting some data about these variants. It's like OMIM, entire ID has data, ClinVar ID has data, NCBI gene description has data. Okay, let's check what we get. Well, 9,000 variants. It's still kind of a lot of variants to analyze, at least now, to do it quickly. So which options we can use? If we are doctors, actually we have to look to all of them, but for the first step, we can choose here end. When we choose here filters to end, what does it mean? It means that all these predictors, all these annotators show this variant as a kind of pathogenic variant. Oh, and then again, we will see what happened. Yeah, we have just 2 variants. So, okay, that's not too long to check. We can apply this filter. So then again, wait a little bit until your variants will be analyzed. Okay, it's done. So we are choosing just variant tab and see what we get there. View table. So we have 2 genes, POMT1 and PNLIP gene, which were found to be pathogenic according to multiple prediction tools like CADexome, ClinPred, SIFT prediction, Reveal prediction, and then we can just see what these variants are responsible for. So we can choose, for example, POMT1 gene and view detailed plan. So we can see ClinVar. This variant is found to be pathogenic. ClinVar ID, we can use it a little bit later. So we see this gene is responsible for muscular dystrophy, type A, muscular dystrophy, dystrophinopathy, congenital with brain and eye abnormalities, type A. So type A. So it kind of correlated with our, I can't see the AMIN here. Maybe I haven't downloaded it like, okay, it's kind of correlate with our clinical symptoms. So we can just put here. And go to the ClinVar. So you see this variant is pathogenic variant. And you can also see which submitter has downloaded the genome way was found as pathogenic. You see that CGAD Center of Human Genetic Tübingen has downloaded submitted that this variant is pathogenic, and it happened in 2021. So it's happened. This variant has been shown to be pathogenic a little bit later than this than the time of which this woman has her genome sequenced to be done. So we can briefly go again to the presentation. So it's what we do. So when we have too many variants, like we got there like 9,000 variants to analyze at the first time, too much to analyze fast, we can go by several ways. First, we can do like we have done, change OR to AND and see the most relevant genetical variants estimated as pathogenic by all predictors and check others. Or, and which is done most frequently, we can add gene list to the filter according to the problem, AJ muscle weakness. So for this clinical case, we tried the second variant and it worked for us since it was not our task. It's what we have done. Was not our task to get an exact diagnosis. The question was just whether she should to resequence her genome, and actually he should, because the quality of the coverage was not that good. So we have found two genes according to our filters: PNLP gene, deficiency of pancreatic lipase. If we can go to the second one, we will find it. So as you remember, she was complained about some gastrointestinal disorders. So the absorption of fats may be impaired. And POMP1 gene is responsible for muscular dystrophy, for dystonia, and defects in this gene are a cause of Walker-Warburg syndrome and limb-girdle muscular dystrophy. So exactly as the complaints that she has. So it's what we see when we go to the clean bar. And then we can slowly go to the clinical case number 2. It's 94306. You can open the result viewer. I will close all the other tabs. Or maybe in that case, it will be a little bit faster. Okay, so until it is downloading, we can talk a little bit. Okay, let's do it. Okay, clinical case number 2. It was a girl. It is a girl. It is a girl just 4 years old. Which indications she got? She has short stature with skeletal dysplasia and delayed psychomotor development, delayed growth. So she's like a little one, very nice, but very little. Low weight gain, and she's not talking. She has delayed speech and language development. But actually, she has some face features which are not usual for like normal genomic ones, muscular hypotonia, etc. And she was providing a test, whole exome sequencing by Illumina, by genetic laboratory in Germany. So until we see the results of the sequencing, we can try to find out the reason for this phenotype by ourselves. Okay, I will also send you. Okay, so It's still working. Open cravat still working. Still haven't analyzed. So I'll just briefly look. Okay, I have sent you a link to the folder. Don't miss it. You will also need it for some more things to download from it. I think the last one is the slowest one. Okay, while it's going, you can download from the link I have sent you the file with list of genes related to intellectual disability. See this file, intellectual, or maybe you don't see this file, intellectual disability file. You can just download it and then, or just open it and then copy paste the genes from there to the field. Okay, it's still analyzing. After it have to be faster, I hope. I don't know, maybe a lot of people get connected, so that's why it's a little bit slowing it down. I don't know how it works at your computers. I think it's also kind of slow. Okay. Okay, so we can just move to the presentation and see what we will do next after the analysis. Maybe I have just to do it one more time. Maybe it will be faster. I don't know. Okay. After it will be downloaded, what are we going to do? Then again, we will go to the filter tab and choose filters, or we will use actually the filter you have already downloaded, the pathogenic one filter that we have already applied. Actually, it's quite universal filter, and you can use it quite broadly when things come to some pathogenic variants analyzing. But we get kind of a lot of variants according to that filter, and I will go further and tell that in this case, changing or to end and analyze by all clinical significant predictions didn't work. So we'll just use the list of genes, which you can download or just copy paste, and we will put them. These genes are related to the pathology, so it's like intellectual disability list of genes. We will put the list of genes to the filter tab, and we will analyze just these genes. It just doesn't wanna work. Okay, I will try to do it on the server. Maybe here it will be faster, so I can just briefly show you how we will work with it. And after it will be downloaded on your computers, you'll be able to do it yourself. Oh, okay, I think here it. Okay. Yeah. Here it's okay, and here it's still analyzing. Okay, I will move to our server to show you fastly. So we are uploading the filter pathogenic with name pathogenic JSON. So, and trying to apply this filter and see that then again we have quite a plenty of results, more than 6,000 results. Actually, we can analyze all of them or just try to use a set of genes. Now here I'm just copy paste the genes from the list. Actually, this list of genes, they are easily found and they exist for different pathologies like cardiovascular, neurological, etc. And all of the commercial laboratories, they analyzed your genome according to the phenotype using gen lists. Firstly, and then if nothing is found, they just switch to the to broaden the search. Okay, 160 variants, so we can retrieve our data. Switch to the variant tab. So still kind of a lot of genes that we have to analyze. And actually, if we are doctors, we will have to take our time and analyze all of them. But now we can do some interesting things. You remember I have talked about the CAD exome. It's a potential tool for pathogenicity prediction, and you can just make your list of genes in accordance to CAD exome score. It's quite easy, and you can start analyzing from the genes which are thought to be most pathogenic according to the CAD exome score. So you can see here that the first one from genes which we can use for neurological diseases, intellectual disabilities, abnormalities is SRCAP gene, and its score is 39. It's quite a high score, so we can find the additional information we will need for the analysis. If we are doctors here, the most important one of the most important is zygosity. So some of the diseases are affects you just when they are in homozygous form. So they are recessive diseases. And some of diseases affect the phenotype even when one allele of the gene is changed, even in heterozygous type. Okay, so we can switch into the detail analysis. View detail plane here. And see ClinVar analysis and AMIM analysis, etc. So ClinVar, it's thought to be pathogenic. ClinVar ID, we will move to that. It's floating harbor syndrome. What does it mean? We will find out from AMIM. I still don't see the AMIM widget. That is strange. Usually it's here. But okay, then we can go just to the ClinVar ID and check it, and go to the AMIM ID and also check whether it's related to our syndrome phenotype. So you see ClinVar ID, it's sort of pathogenic, floating harbor syndrome, autosomal dominant inheritance. It means that even one broken copy of this gene, like we have in the patient, is enough to develop the phenotype of the disease. Still, here it's still working. I don't know. I hope at your computers it's a little bit faster. So let's move to the presentation again. So what we have done, we have applied a set of genes related to our phenotype, then applied the filter, which we have already uploaded previously, got some variants. Then we made sorting according to category pH-red. pH-red, what does it mean? The last column of the provided files is pH-red like. It's the value greater of 10 indicates that these are predicted to be one of the 10% most deleterious substitutions that you can do to the human genome. So and the score greater or equal 20 indicates 1% of most deleterious and so on. And we got 39, it's a higher score. So then we go to the CleanVar widget and see that it's pathogenic. Sequence ontology is top gained. A sequence variant whereby at least one base of a codon is changed, resulting in a premature stop codon. So it's quite deleterious thing. CleanVar say that floating harbor syndrome, Provine, Reveal, and Sift has no data. So that's why we couldn't just check or to end like we made previously and check all of the pathogenicity scores. OMIM, it's ID for amine, zygosity, heterozygous. So if you go to the amine data, you can find it's even heterozygous mutations. It's exactly what we saw in the genome of this patient. In this gene also cause development and delay, hypotonia, musculoskeletal defects, and behavioral abnormalities. It's a rare genetic disorder which is characterized by short stature, delayed bone age, delayed speech development, and typical facial features. Yeah, so it's exactly what we were looking for. It's exactly in accordance to a phenotype of our patient. And when we go to the CleanVar, we see that interpretation is pathogenic and autosomal dominant inheritance. What if we will look it from 6,000 variants, if we wouldn't apply the gen list? We will anyway find this variant, so we will have to check from the very up all of the genes, which pathologies they are related to, how do they inherit it, whether they are in accordance to our phenotype or not, etc. And then again, on the 1, 2, 3, 4, 5, 6, 7th place, we will find the gene which are most likely responsible for our phenotype and this disease. So when we write, it's what we got from the CGAT report. So exactly, it's a commercial laboratory which providing genetical testing for the diseases. And they also found, they detected a pathogenic variant in gen SRCAP, which is a causative of floating harbor syndrome in your patient. So you see that I can tell it's quite of a complicated and it's a high responsibility to work with patient genomes, but everything is possible. And I know that when you will work with your own genome, it's not such a responsibility. So you will not look for some genetical abnormalities. Most likely it's just some variants of some traits which are not that pathogenic. So the next step of our work is to work with our own BCF files. Yeah, I will briefly stop it and look. Okay. I don't know whether it will be possible to work with our own genomes, whether OpenCravat will allow us to do it now, but if you will have questions with working with your own VCF files with OpenCravat, you can just ask it directly and I will help to choose a set of filters, a set of annotators, etc. Okay, so then again, let's move to the presentation. Yep. So let's briefly check some things, some common things which can be analyzed in your genome. Or in my case, I will analyze in Anton's genome because I haven't already get the results of my own genome sequences. It's methylation, folate metabolism, need of supplementation. It's a very popular thing. Neurotransmitters, CHRNA3, smoking adduction, and everything you want to check afterwards. You will be able to do that. So methylation, what is that? What's the methylation genes, folate metabolism genes? If you're related to kind of longevity, personal medicine, you can't hear about that. It's a very important cycle in our organism, which makes possible some chemical changes, and it affects, it includes methionine metabolism, methionine to homocysteine transversion. And if this cycle, if these methylation processes don't work appropriately, it can lead to several pathologies, like you know that appropriate gene methylations allow silence some genes. For example, oncogenes are silenced, retrotransposons genes are silenced, and it's a problem when with age, methylation kind of changes, and some retrotransposons became active, and it affects our genome stability, and also and actually it's one of the reasons of aging. But then again, about methylation, and let's go back to it. Appropriate work of methylation cycle enables appropriate toxins clearance, glutathione system work, mitochondria appropriate functioning, because of during that cycle also glutathione is formed, etc. And this cycle is really dependent on vitamin of B group availability. Yeah, so you obtain them from our diet, like B12, B9, folate, B6, etc. But some people have gene variants which makes availability of these vitamins like lower. So they are transformed to their active forms, in which they are going to the metabolic cycles in a less extent than like people with different genetic variants. And actually, it's corrected quite easily. If you know your gene variants, what is affected, you can use some kind of supplementation. Usually, if you have changes in MTHFR gene, and okay, gene changes is not a prescription for taking something, but it's a kind of an alert that you have to, maybe you have to check your blood level for these vitamins, and combining such results, make a decision with your health practitioner about supplementing or not supplementing some vitamins, or maybe increasing them in your diet, etc. So MTHFR gene, it's B9 usually, MTHFR, it's cobalamin, B12, CBS gene. It's very interesting gene. It depends on B6 vitamin, and actually the working of this cystathionine beta synthase, it gains, it is related to serum metabolism, which is also very important for mitochondria functioning, etc., and the BHMT gene to beta. So if you have already your genome uploaded, or you are planning to do it a little bit later after our workshop, you can use it with filters I have shown that we usually use, annotators, I'm sorry, and filters as well. And we can just check these genes. So I will try to work on Anton's genome and hope that Open Cravat will work now. So I'll just open the result viewer. Yeah, okay, it works. Variant, filter. Then again, we have too much variants and we need to apply filter. So we can upload our favorite JSON filter and it will show whether some kind of deleterious variants we have. I will copy paste the genes from the presentation. Okay. Or you can just type them like MTHFR, MTR. I will send you a list of genes so you can also use them for analysis. Okay, and we will see what we get. Okay, we will see how many variants we will have. Okay, just 3 variants. It's nice. Apply filter. Okay. Variant tab, view table. What we see? Some genes, MTHFR gene, just MTR, and again MTR gene. Okay, we can go to the right and see different annotators, like what tells us. Linvar, methylcobalamin, it's methylcobalamin metabolism is affected, etc., etc., etc. From GKB. Okay, shift prediction, damaging to related damaging. Okay, but when we see it, we should definitely see the zygosity of this gene. When they're heterozygous, it's not that that much affected by your vitamin metabolism, like when it's homozygous. Oh, then we can see, for example, we are choosing the gene, we're analyzing MTR gene and show the detailed plan and see what this gene is responsible for. Oh, you can see methionine biosynthesis have been identified like a cause of methylcobalamin deficiency. Yeah, so if you have some variants of this gene and you see it can be pathogenic, you can choose it by, you can check it by different annotators and see what do they say, methylcobalamin deficiency. Also analyze some papers from PubMed and find out that, okay, I need some additional supplementation of vitamins. For example, if we are talking about these genes. Okay, let's go again to the presentation. Okay, it's what we have gained. So we have found several polymorphisms estimated as damaging. They're associated with homocysteinuria. It's when your homocysteine is not converted to methionine appropriately and its level increased, and you can see it in your blood test, and it's a popular and famous, like important marker of cardiovascular diseases risk. And it can be corrected if there are some problems with imbalancing of methylation cycle disturbing. If you also will download the annotator for the drug interaction database, you will see that cyanocobalamin affects this gene. So usage of cyanocobalamin may help prevent the negative impacts of this gene variant. But then again, we should definitely check the blood level for that stuff, because genes are not that easy as we sometimes think. So the MTR gene, it encodes the enzyme which is called methionine synthase reductase, which enables MTR to turn homocysteine into methionine. But for this, we need enough B12 vitamin. if the enzyme activity is low, the risk of increasing of homocysteine level and cardiovascular diseases is increasing. Yeah, and polymorphism in these genes are associated with higher needs for vitamin B12 supplementation. So you should check your status of these vitamins to prevent some negative outcomes. And actually, folate metabolism genes are very popular in different commercial sets of analysis you can see, and some of them are quite expensive. So they will just tell that we will reveal your folate metabolism or methylation, I don't know, status, etc. And actually, you can do it easily just by analyzing a few genes. You have a list of them by uploading the filter and just looking to the information from the open cravat. Then what else we can analyze? We can analyze some genes related to neurotransmitters. For example, COMT gene, it's catechol aminotransferase, and the product of this gene, it affects noradrenaline and dopamine cleavage. So the product of this gene, it breaks down dopamine and noradrenaline. HTR2A, it's a receptor of serotonin. And you know, there are popular medicine like inhibitors of serotonin receptors and activators, etc. And DBH, dopamine beta hydroxylase. So when we are analyzing this gene, what does it affect? It affects our behavior genetics, like we can make some predictions about our motivation, etc. The most studied thing is risk of neurological disorders according to these gene variants. The need of supplementation, maybe magnesium, tryptophan, etc. And antidepressants usage. So actually, it's also related to pharmacogenetics testing. These polymorphism of these genes are related to changed response to some groups of antidepressants. Okay, so we can, again, go to Open Cravat, put these genes into the filter tab and analyze which variants do we have. I will do it now. I will send a list of genes. It's just actually 3 of them to our chat room. Oh, I'm sorry. I did it wrong. So just 3 of them. Okay. It's Anton's genome. We're just putting the variants and then see what we get. Sometimes a table sign disappeared here, so you can just go to the gene tab and choose view table, and then it appeared. Okay, but when you're what I want to say, when you're analyzing some genes which are not related to any pathologies, you just want to know about some variants you get, you can just try to clear filters and just see what substitutions, which changes you have in that genes. So just without filters, just type and see what you get. So we can see plenty of changes in HTR2A gene, COM gene, etc., etc., etc. And then we can just look for the thing we are interested in from, for example, some scientific data, etc. Like we have, some interesting stuff from genetics. According to COM gene, it's like a warrior or warrior model. It's a gene variant that has been shown to predict the ability to maintain cognitive ability during competition, etc. I will try to find this gene variant. Wait. And according to that, you can make predictions on your stress response because people with one genetic variant, which has, okay. Quite a plenty of substitutions. So let's go back to the presentations. You see from Anton's genome, okay, slideshow mode. Okay, genotype that he got, GA genotype, it's valine to methionine substitution. If we have two alleles of methionine, it means low activity high dopamine allele carriers outperform like unchanged variant on a variety of cognitive tasks, because in like a normal state, we have high dopamine level, but it reverses under stress conditions, and homozygous change, which related to high dopamine and low stress resistance, high cognitive abilities, headaches may occur. And if we know that, we should not use precursors of neurotransmitters, etc. So what else we can do by using the open cravat? How to use in daily life with your genetic data. So you're just reading some interesting articles. Came across an interesting article. For example, there was one recently about leadership. So leadership, sample of bosses according to UK Biobank were analyzed, and they were found several variants which were associated with leadership phenotypes. With a focus on holding a leadership position and performing critical managing functions, etc. So you can find in this article this list of genes, a set of polymorphisms which are associated with this stuff, and you wanna find out whether you have some abilities to be to gain a leadership position or something like that. You can't find it if you have already done your genome sequence, because this article may just appear and you haven't this data yet in commercial analyzers. So you can just copy the gen list you want to analyze. Then again, go to your genome, put it into the filter tab, and okay, you have some kind of plenty of results here. And but anyway, you can analyze them and check it. So, but you also have here the RS IDs which are or are not in this article. And having them from your article, you can just check whether you have these substitutions or not. So kind of that stuff. So maybe you're not having or having them, etc. So I have analyzed Anton's genome and have found that he has got several RSIDs related to these leadership genes, but just 2 of them. And if you find that you don't have any, you don't have to be upset, because it was observed that some positive genetic correlation between leadership position and some negative well-being indicators, including high levels of bipolar disorder, etc. So maybe the 2. I don't know why haven't I found it now. Definitely have been there. Okay, just I will check it one more time, but I was sure that it was there. I was checking it several times. So you can just put a list of genes, put a list of RSIDs you want to check, and then just check from some interesting article whether you have some interesting stuff from that or that you are not. And maybe one of the last genes, like some new information is always appearing in the literature. For example, ChRNA3 gene, it's related to smoking addiction. It's coding the nicotinic acetylcholine receptor. And several negative traits are associated with this gene. Nicotine dependence, cigarette consumption, smoking cessation, lung cancer, and some chronic diseases of lungs. So, and you want to just check, are you lucky to not have these variants, or you're not lucky and you have this variant? So you, as usually, you should just easily check this gene by putting it into the okay, we have to open. Filter genes, just put a gene. Okay, apply filter. It's Anton's file. Yeah, file. Well, it's kind of slow. Until it analyzing, I will show on slides what we got. Okay, so we have found that Anton indeed is a carrier of some negative change, that like this RSID, and indeed it was associated with this negative impact. So obviously he should not smoke. And you can easily check it on your own genome. And the last gene I want to talk about, you can also check it on Anton's genome. Okay, and I will briefly tell how to influence it, that gene. I will try to do it in our server because it's, I think it will be a little bit faster. So usually it's not that slow like now. We can use our preloaded filter. Use the gene we want to analyze. Apply it. and see whether we have some negative substitutions or not. So you see that according to the Open Cravat, indeed, it was found one variant of this gene, which is homozygous, so it affects more severe, which predicted to be damaging in blood gene. And what does it mean? You can find some information from the detailed plan from the widgets of different annotators. Depends on how many annotators you have used. You can see that this gene encodes component of pyruvatic hydrogenase complex, etc. And you can find all of this information here and go to the links, etc. But I will briefly show you on the slides what does it mean and how can you just influence it. Okay, so then again, this gene is responsible of transport of pyruvate. This gene encodes an enzyme, E2 part of complex in pyruvate hydrogenase complex. It's a part of a complex that plays an important role in the pathway that converts the energy from carbs into a form that cells into the form of ATP energy. So here you can see this complex, which transports carbs to the mitochondria, and they will make energy for us in ATP form. Carbs and fats are the main energy sources for our cells, for our organism. And for carbs transporting into the mitochondria, responsible this pyruvate hydrogenase complex. And if there are some problems with it, especially, for example, in this case, in E2 subunit. Okay, oh no, I haven't completed yet. Yes, it really depends on lipoic acid, so we should probably supplement it and check it into the level of it, etc. And if you are a carrier of such a variant, you may suffer for some weakness, for some faint when you get up, when you stand up quickly, you may feel dizzy due to this gene changing. So you should pay attention to balanced diet, to carbs and fats, etc., and maybe supplement with lipoic acid. But okay, individual genes are important for different stuff, but actually there are just a few genes which we can tell like confidently that they're indeed important, even if you find changes in these single genes, like top important genes. Genes are related with each other. They interact, they form gene nets. So if you wanna really discover whether this trait is about you or not, you should analyze like a whole set of your genes. And for doing that, we use polygenic risk scores. And I am happy to give talk to my colleague who will tell about polygenic risk scores. Okay.
Hi, everyone. My name is Alina, and I will talk to you about polygenic risk scores. And I'll just share my screen. So thank you, Olga, for a nice introduction. And yeah, now we have already know how we can work with single polymorphism, but it's probably not enough just read the descriptions of genes or polymorphism. You need to have some model that can predict phenotypes. The first thing that you can presume, okay, I just get the coolest neural network possible, the best architecture, and you get some interesting predictions. But when you start dig data, you realize, firstly, it's extremely hard to get access to the data, in biobanks. Everything is so bureaucratized. And secondly, that you discover that this data can occupy a lot of gigabytes, but the number of samples are usually smaller than the number of features. So how you can see in this slide, you can construct some matrix. In UK Biobank, it's about 500,000 biosamples. In Japan Biobank, 200,000 samples. And features, it's about 850,000 variants. So you have matrix where the samples as rows and features as columns, and you can make some prediction where there is health, where there are disease.
Ce qu'elle a dit. Ouais, elle a remis Cohen en place l'autre jour. Hein. Il a couplé en plein milieu de phrase. Mais j'ai dit, laissez-moi terminer. Virgule.
Okay. And but this matrix disbalanced in terms of number of samples and number of features. And it's extremely easy for model to overfit. So overfitting is when we have good performance on the training data and poor generalization to other data. So, and another problem that we have far more variance than we directly measured. So it's towards 90 million variants imputed. So imputation is a kind of statistical inference of unobserved variants. We do it by comparison with the reference genome. So why we want to predict something, why we want to build some machine learning models. With that models, we can predict complex traits. And with that kind of models, we can facilitate disease screening and prevention at the population scale, aid in the development of personalized medicine, improve symptom diagnosis and intervention at an early stage of diseases. We can reconstruct the genetic predisposition of an individual to a particular disease, and also we can apply it to the breeding values of animals at some kind of genomic selection. And what is polygenic risk score is an estimate of an individual's genetic risk for some trait obtained by aggregating and quantifying the effect of many common variants. So we can see this simple formula. We just summing up some product of beta is the effect size of variant. We can just get it from GWAS analysis, some statistical analysis of your data. Multiply by dosage, number of copies of SNP and the genotype of individual. How we can calculate dosage? It depends of the method you use to infer your SNP. So if you directly measure it, we SNP will have probability 1 of one genotype and 0 for the others, but at an imputed SNP, an individual might have, for example, a 90% probability of being homozygous for allele A and 10% of probability of being heterozygous. So how we can turn from this simple formula, which we already know for PRS, to machine learning models? Just through linear regression. We all probably know this simple formula from our school years. It's just linear regression, where y is an in vector of the phenotypes. So left part is phenotypes and right part of equation is genotypes. X as an n by p matrix of genotypes collected across p's and p's on the same set of individuals. Beta is a p vector of SNP effect sizes, and epsilon is an n vector of residual errors. Despite its simplicity, the above model is instrumental for understanding almost all existing PRS methods. But we can keep in mind that we have unbalanced data, so we can solve this problem with applying some kind of regularization to our data. So it's just trying to shrink coefficients in equation to 0. And how can we actually predict new observations through PRS construction? We can predict phenotypes for newly observed individuals using the estimated SNP effect size beta from the modeling equation. Specifically, once we have obtained the p vector of genotypes XL for a new individual L, we can simply plug in the SNP effect estimates to obtain the predicted phenotype value, PGS, as in that way on the bottom of the slide. And we can explore some existing models. So some scientists have already overcome those difficulties with bureaucratized biobanks and with overfitting models, and successfully applied some models to the data. And we can see that some model with regularization, lasso, elastic net is the most efficient models. We can understand it just by R-square coefficient is a determination coefficient, which tells us how good is the model. So as closer to the 1, as the better the model. So when we explore our own genome, we can find some polygenic risk score. We can find it free on the internet. There are 2 ways. We can just find paper for topic we're interested in, or just find hosted and curated scores. For example, polygenic score catalog, PGS catalog, and cancer PRS web. They're very convenient way to find PRS and information from the article, from the sample, or about the samples. And about some specifics of PRS, we should keep in mind that the larger datasets give more power to detect association of SNP with the trait of interest, and have been shown to yield scores with higher predictive capability. And the PRS will perform best in population of the same ancestry as those in which they were derived. And finally, when using an existing PRS, it is important to first ensure that it is of good quality and is appropriate for the analysis data. We can find this information just in papers. And generally, we need to know what kind of information we need to find in our waiting files of polygenic rescores. At a minimum, the information needed to replicate the PRS is a list of SNP, so with some RSID or base pair positions on chromosome, the effect allele for each SNP, and the effect size for each SNP for the condition of interest, and the genome build. And I have pointed out that training models is extremely tough and bureaucratized. So far, we decided we can use already trained models PRS with published coefficients to provide output to our users about their disease risk. And Alex Karmazin will talk more in depth how we do it now. Thank you.
Hello, I'm Alex Karmazin. I'm software development for the Just DNA-seq project, and now I will explain a little bit our architecture and how we use PRS with OpenCravat. So nowadays, there is plenty of PRS on the web. You could use this catalog and just download a PRS as a text file, and then you should do something with it to get results and helpful information. In our case, we use the PRS for the personalized reports that we developing now. So the general idea is that we have the PRS text file. We make a parser that gets this data and creates a SQLite database. Then we create OpenCravat or OCravat. It's the same annotator. It's just a piece of Python code that have some function that triggers by OpenCravat each time it has the new SNP, and we just make a query to our database, and if we have some data, we could just put this data in the table that we see on OpenCravat. In our case, these data will be the weights that have this PRS that we are using. And here we have some trick that we have different versions of the VCF file, and the PRS could be trained on one version, and our user could have some different version. So that's why it's easier to stick to such arrays that have arrays ID in it. So in this case, we could use the some other annotator DB SNP to get the RSID, and then we could query our database not with the chromosome position alt and ref, but with the RSID. Then we will have a column with weights in it, and then in different piece of Python code that will represent our reporter, we need to get weights from our PRS column and from the zygosity column, and then we could make weighted sum, and it will be our PRS value. So, and let me explain some our next steps that could be interesting for you in the way that you could use open cravats in several different ways. So first, if you work with PRS, sometimes you have a lack of information how you could interpret PRS values. And in this case, at least you could have percentiles and see how unique your data is for a particular genome. And for this purpose, we plan to use the GNOME-AD database. It have frequencies for the 8 populations and global frequency, and we could just have a generator script that would from one side get the PRS data, and from the other side would have frequencies for the particular SNPs, and this is enough to sample the data, and then we will have a normal distribution, and having sigma and mean, it's enough to have to calculate percentiles and all other information that we want. And we could go even further, and it's our next step. After the next step, we could identify a population of particular genome, and in this case, again, the GNOME-AD database will help us. So in this case, we could use a Bayesian rule. So first, we will have a uniform distribution for all 8 population, and go from one SNP to another. We will update our prior probabilities. So for example, for first S&P here as the prior probability, we will have 1/8 as a uniform distribution. And for example, we want to know the African frequency. So we would get the data about the African frequency and put it here is a probability of getting this SNP given we have African probability, and we will divide it out at the global frequency. On the next step, with the next SNP, we will take this probability as our result from the previous step and plug it as our prior probability and get the P given A and PP from our database for the next SNP, and we could proceed this process for the each population until we will have a probability distribution for this particular genome. So in this way we could tell whether you're African, American, Swedish, or someone else. So this is our plans, and we will investigate these possibilities with OpenCroat. Thank you, and now Anton will continue. Anton, you could just unmute your mic.
Yeah, I am doing this. Yeah, thank you very much for very detailed explanation. Actually, I want to highlight that population identification is a huge headache, and it's important not only like for general curiosity to know whether I am a Jew or whether I am more European or more Asian, but also to correct results of polygenic risk scores. The problem is that, as Alina mentioned, because of a huge number of features and smaller number of samples, we have great overfitting. We fight with overfitting using different regularization and cross-validation techniques. But still, in classifiers and regressors, we get a lot of false positives, and we also get a lot of covariates. So when you select the genes that are most predictive for some phenotype, some of genes are causal and some of genes are just covariates. And the problem is that it's very hard to define it. And usually when most of the classifiers are trained on some specific populations, and they include variants that are specific for this population, and they take into account peculiarities of this population. And when you apply the same model on another population, you have a reduction in accuracy. And if the user uploads her genome and applies some predictive models to get phenotype predictions, she should get some warnings for populations that are far from hers, to take those predictions with a grain of salt, because they may perform worse because of differences in population. But of course, it's not the only plans that we have. So now I will talk a little bit about future plans. Actually, when we started the project, we had two major motivation. First, we were unhappy that most of the commercial services, even though sometimes they give nice reports, they are often not explainable. So we don't know the way how they actually came to conclusions, especially when they have some risk scores. And they also don't provide flexibility to start from some existing reports, but then do some manual investigation. And in the same time, platforms like OpenCravat and Oakwire, they have huge flexibility, but as people mentioned, their user interface was made for people like Olga, who are professional medical geneticists, and they have specific clinical problems in mind. So you have a lot of filters, but you know what you want, and you just play with them. And what we really want, we want to join flexibility of existing scientific and medical solutions like Oakwire with something user-friendly, when you have some predefined filters that you can change that give more or less optimal selection for major use cases. Like, for example, at this workshop, you applied some standard filters to find pathogenic variants, and you had a bunch of conditions that were good to start with, and then you just increased or decreased filters. And we want to also produce nice looking reports. So far, we thought about longevity report, and also a report about major risks for your longevity, because as you noticed, heritability of longevity is not very high, but when we make decisions about our lives, we actually can get some useful information about our risks and take action. Like, for example, with statins, I probably have to take smaller dosages if I will need to, because I can hurt myself quite seriously. Or like with B vitamin deficiency, I already bought some vitamins, B12, because I have issue with this. I still have to check my blood, but it's just like an example you dealt with today. And we also want to look for important things like if we have any adverse reactions on some drugs, especially some drugs that can be taken either like statin as part of your life, or as metformin, or as rapamycin as part of your attempts to live longer. So if you think maybe, like many of my friends actually do, they start taking metformin because there are many papers, and then some of them see that it's not for them, they have some unpleasant reactions, and it's actually better to know from your genes maybe this life-extending drug would actually hurt you. And of course, when you decide about drugs, you probably still should talk with pharmacists and doctors, just showing your genetic report to let them know, but better trust professionals to help you. And we also want to integrate what is not there in OpenCravat. It's polygenic risk scores, and we want to make some reports with them. And of course, as we also want to contribute to science, we plan to publish at least one paper on it. And with current budget, we have a plan for 1.5 years. And so far, I don't know if you realized, but from getting the funding, we are active maybe only 4 minus 1 month. So originally it was just weekend activity of me and Newton Winter, like for a year when we were like writing the pipelines. So that what we were mostly doing before we got Gitcoin funding. And when we got Gitcoin funding, half of our team had to evacuate relatives from Ukraine or to evacuate themselves, like Olga. So we are kind of pretty early in development, but in the same time, as you see, we are making workshops. And the reason why we are doing it, because we believe that in open source projects, community is the key. And we want to establish relationship with users and contributors as fast as possible. That's why we are really interested in your feedback. So we want to talk with 1/3 of you who already sequenced the genome, and also those almost 2/3 who plan to. We also want designers and developers to contribute to our projects. And we also think that contributing to OAK-VAR, because we are building on top of OAK-VAR, it will be also quite helpful. And we also need some help, because we are actually extending longevity map to make longevity polygenic scores and longevity SNP module. And there, for example, longevity map was last publicly updated in 2017. So people who want to contribute and have biological skills to read papers are also welcome. And we are also happy about everybody spreading the word about project. And I also want to give you some links that can be useful for you to participate. So like internationally, if you are into longevity, you can join some Discord servers like Longevity Fellowship if you want to be involved as activist or entrepreneur or researcher in aging research. If you want to help aging research projects get funding, maybe joining Vita DAO and other DAOs that are valid and that are doing real things can be useful. So you can check their Discord. And we are also happy that we have some people from gerontology research group mailing list here, and also some people from Lifespan.io, from DNA Deviant and other communities. Also, of course, you can check our Twitter and GitHub. And as we have some people in Romania, and we are happy to collaborate with Romanian Bioinformatic Society. I also recommend to check RSBI website and events. For those who are in Romania and interested in longevity, we have a meetup group, and we also have Facebook chat where we exchange. So all the information will be also sent to you after the workshop, together with the link to the presentation. And now we are finishing the main part of the workshop about personalized genomics, and we will finally have a Q&A session, maybe like for 10 or 15 minutes. And after that, those of you who have interest in gene editing and are not very tired and want to learn it, we will have a short bonus track to actually learn how to design CRISPR, both regular CRISPR, epigenetic CRISPR, and I don't know if we will manage to design newest CRISPR paste to actually fix it and to pack it in some viral vector. So thank you very much for your attention, and we'll be happy to answer your questions. Please write your questions, and any of the team can answer it in voice. Guys, don't be shy. Also, if you maybe have not a question, but if you have suggestion, what can be changed, what can be improved, or how we can collaborate more, also feel free to share. Yeah, also thank you, Daniel, for attending. Okay, guys, so we will wait 5 more minutes. If nobody will ask anything, we will start the extra module.
Is there a Q&A session after the extra module, if I may ask?
Sorry, Mihai, can you speak louder, please?
Yes, of course. Is there a Q&A session after the extra module, if I may ask?
Yeah, I mean, so right now you ask questions about like what you learned, and also you can have maybe 5 minutes break, and then there will be a very short module on editing, and after that, of course, if anybody has any questions on any topic, we will answer.
Excellent, thank you.
Regarding Johnny Adams' question, actually, maybe you can ask it out loud, Johnny. Unmute yourself.
Hello. Thank you to all of you who made this exciting presentation. It's a glimpse of the future. I would like to hire a trainer or a coach or someone to teach me, maybe others, on personalized consultations on how to use these systems. Probably best if you send an email to jadams@grg.org, jadams@grg.org, or if you'd like to speak and tell me now and give me your contact information in the chat window. Thank you.
Yeah, thank you. I think it's actually an open question how to practically use it, because I mean, we are still developing it and we are collecting more data to prepare the reports. I think maybe the best person out of who I know, maybe you can talk with Olga and she can give you some advice, but it's a work in progress. We will have to do some additional curation because it's a lot of work to summarize latest discoveries. We are now having headache summarizing what already exists, like adding additional fields to longevity map. And yes, so for this, maybe you can talk with Olga, and for technical stuff, if something has bugs, you can open an issue maybe at OAKVAR. Olga, maybe you can have some ideas.
Well, yeah, it's such a complicated question because we're still just on the beginning of our way to understand how can we do age management. So based on the things we already know, I can explain how to use OpenCravat for searching for gene variants and for some stuff for age management that we already know, but it's just such a small bit of information that are relevant and we can rely on for it. So it's indeed kind of an open question. We can discuss it a little bit later. I will think about your question and what can be done and in which way it would be to do it the best.
Well,
go ahead.
Okay, so I just also wanted to add that in the lab, we also are going to open source code for other omics stuff. For example, right now, sequencing of RNA is pretty cheap, even though it needs some improvement to switch from sending blood to maybe sending blood in a medical way to sending blood and specific devices to avoid like cold delivery. But we also plan to share some code with RNA related data where you can probably run something like a bit-H RNA clock, but it's something of lower priority. What I am actually a big supporter is that we should push some efforts to develop more open source tools and open source ecosystem. It's better like if major part will be opened, and if people want to earn, it can be done through consultancy or through some very specific features.
I wanted to ask, so do you plan to build some profitable business from it?
Well, so far we want to, I mean, we have a lot of tasks to make it like open source, and we have like a lot of features we want to develop. I mean, we don't have plans right now. I believe in case of Rick, he will probably develop it as a platform and will sell additional services, but he is our advisor, but our team is a bit separate. And for our team, we plan to deliver what we plan like for a year and a half, what we presented to you. So as soon as it will be built, maybe we can think about any types of side projects, or maybe not. What we really want is to keep the core stuff totally open source, because we believe such information should be open so others can improve on it. And for Rick, actually, maybe for Okwar, he can, maybe Rick, maybe you can answer for Okwar. What are your plans there?
Hi, everybody. I have a question. Maybe I miss it, but I didn't listen to everything. Is there any score for polygenic score for longevity? Because as I seen, your interpreter only shows the presence or absence of some gene. But what about how to quantificate this?
Alex, so far, yes, there are some polygenic scores for longevity. I think I forgot to cite that paper in my part for some reason, but they are highly inaccurate, and we have to validate it and implement it. And our plan is that we started with like lower hanging fruit, because like getting polymorphism first is easier, and next step will be implementing polygenic scores and validating them. Also, we are talking with João Pedro if he has access to UK Biobank. Maybe there are ways to actually train some models to get our own models, because they don't really have good distribution of long-living people there. But most of like participants there are like not older than like 60, 70, but they have parental longevity. That means that you can train a regressor that can predict parental longevity based on genotype, and say predict, for example, what is the chance to have a parent that lives longer than 90 years old. And this probably can be trained. The problem is that we really lack training data on, I mean, there are some like studies published on centenarians, but if we talk about thousands of samples, we don't have thousands of samples of centenarians. And for UK Biobank, it's mostly wild type people for which we don't know, we don't have many centenarians, unfortunately.
Yes, because it's an eternal question about validation, and there are so many uncertainty, and there are so many controversial in different population when we're talking about longevity, and it's a good idea to validate it. Anyway, I remind you that we have our own sample of maybe 200 centenarian from Ukraine as well, so we can validate your future.
Well, are they sequenced, Alex?
Your forthcoming score on this sample.
I think that if they are not sequenced, maybe we should first get funding to get them sequenced.
I'm in process about this.
Yeah, it's nice. Also, we'll finally have more of, you know, because it's so like British and Japanese and Finnish centered. Maybe we need more long-living Ukrainian population as well. I don't know. Like we need more data that is outside of just regular biobanks. I think that it's 3 biobanks plus Italians. That's like, and maybe Americans also, but other populations are not that spread in terms of data availability. Yeah, so Alina gave you some links.
Yeah, okay. I have a question for Ola. Yeah, thank you for a very interesting analysis of few genes, but especially about mitochondria. I know it's your big passion, but as I've seen on your tables, most genes, maybe 90% of genes of SNPs, it's in introns and non-coding sequences. What do you think, for what percentage of gene you will be able to make such beautiful stories like MHTP or about uncoupled protein genes and find some supplements? It's 1%, 2%, or 3%?
Yes, what genes, what tables you mean, like? About longevity, our longevity annotator. Yeah, it's showed in a table, the longevity annotator table.
No, I'm talking about MTHFR. It's methylation cycle and uncoupled proteins, and to use, I don't know, aspirin to treat some lack of function of this gene when we're talking about mitochondrial genes. So it's good example. We always use it, but as I see, it's only a few examples there are about very curable genes in this longevity list.
Well, yes, I know it's a problem. It would be very nice if we can do everything, you know, that just, I don't know, I can count these genes, I don't know, with my two hands, with fingers on my two hands, maybe we can affect with such stuff.
Now, what is your estimation? We are talking about 1% or 2%, or maybe since God.
But Alex, you actually forget that there are methods to estimate the effect of non-coding regions, because there are tools and even neural networks that try to, if it is like within enhancer or a similar active region, there are ways to actually compute the chance that it can influence activation of some genes. Of course, it's not really that straightforward as with coding variations, but it doesn't mean that we cannot say anything about non-coding mutations.
Well, yes, it's kind of a complicated question. Indeed, we are focused on several ways. It's not just longevity. There are few steps like lower the risk, so we can just estimate risk of some common diseases like cardiovascular, etc. And you can do regular things for it after you're getting your personal risks, like doing annual checking of blood parameters, some ultrasound diagnostic, etc. So just monitor your risk, and it's an important part in personal genomics you can do. About genetics and longevity, about especially some longevity genes, indeed, there are just very few things we can now affect. It's such a fine-tuned system, and we can easily just break it down. So we have to focus on risk management, then estimate maybe longevity potential. It's more like for curiosity stuff, or maybe in future, like a lot of biotech companies are searching for ways to, I don't know, target some longevity genes to increase expression, decrease expression, etc. So it's like an our future, and it's not exactly the task which would be ours in that project. So we have to just show that you have such variants. Okay, you're a lucky one, or okay, maybe you're not that lucky, but still you have things to do to improve your health, lower your risks, take care of medicine, just pay attention. You have to use this medicine with caution.
maybe you have to use some supplements if there are some genes. Indeed, we are just on the beginning of our way on understanding a lot of gene functions, etc. So, okay, you know, it's not worse than I am.
Okay, guys, I suggest maybe we can switch to a bonus track, and after that, if you want to stay and ask more questions, we will have a second Q&A session. I will share my screen again, and okay, guys, so most of the time we talked about longevity genes, and as well as normal genes, as well as damaging genes, and with practical examples, you saw that there is not everything well in my genome. There are some damaging and pathogenic variants. And in this workshop, bonus workshop, we will talk what can be done if you are unhappy about some mutations. It's a little bit futuristic because you probably want to wait a little bit when the technologies will get more safer and easier to use, but still actually you can pretty easily design gene editing tools, and you can pretty easily pack it in viral vector, and that's what I will share now. So when we are unhappy about our damaging gene variants, we want to fix them with good ones using different gene editing tools. And of course, different types of CRISPRs are the most popular ones. And but please, when you need a doctor to fix your genes, don't go to this doctor. So on the picture is John Kui, the guy who applied Cas9 on 2 twins. Actually, I believe it was really brave of him, but he did a terrible job by using wild type Cas9 that could cause additional mutations and slightly hurt the health. So let's first look a little bit at the theory. And we all know that for CRISPR-Cas9, there was a Nobel Prize awarded to Dudna and her collaborators. And originally, just to remind you that it was part of immune system of bacteria, and they worked with Streptococcus that is slightly pathogenic. And it's the main idea is that you have bacteriophages that, like on this picture, that get into the bacteria, and they have DNA. And the idea of CRISPR is that it can recognize foreign DNA and cut the hell out of it. And you have those CRISPR arrays, and they use RNA as the guide. So it unveins the DNA, matches RNA if it fits. If it fits, it does the cutting. And people, after people moved the system to use it for gene editing on mammalian cells, originally it was just knockdowns when you just make cuts and destroy genes. Then they utilized homology and non-homology directed repair to give a DNA template and make an insertion of the transgene. And actually, even though many labs, including some labs in our institute are using this technology, it's already somewhat outdated. So using homology-directed repair for insertion is actually not the very best technique now, surprisingly. Several years ago, it was really hot. And if we look into CRISPR and gene editing overall, so the idea that you can program some protein to get into specific region and do the cutting is not new. People use talons, people use zinc fingers motifs, and actually talon is still more accurate than CRISPR, but because you had to do changes in the proteins, it was a headache to use. And when CRISPR emerged, everybody switched to CRISPR. And as you can see from this gene editing trials, you see that 63% of them are done with CRISPR because you don't change protein, you just change RNA. And the other important stuff is that mostly they prefer to edit cells because you have a problem of delivery. So you see like in vivo gene editing and ex vivo gene editing. And ex vivo means that we take patient cells, we do editing, maybe we want to grow them, and then we put them back and cells do the job. And it's way safer and way easier to do. So most of the trials, as you see, are ex vivo. And also, you also see that most of ex vivo, like 68%, are CAR T cells. So it's T cells that we want to reprogram to target cancer. So they bind to cancer cells and kill them. And it's for some types of cancer, it's the best therapy ever, especially non-soluble cancers. And in terms of Romania, by the way, I think in Fundeni, they're doing some experiments, and also in several other hospitals and labs with CAR T cells. In our lab, we worked with other types of cells. So we had several cell lines that we actually edited for research purposes. And yes, so you see that the CRISPR is actually leader, and all the time new types of CRISPRs and new modifications emerge. And what actually he did in that famous human germline editing, for which he got actually a sentence. I don't remember, I think like 1 or 2 years of jail time, and he is on freedom again. So he knocked down CCR5 gene in Lulu and Nana, and they should be 3 years old by now. At least they were healthy. I don't know if they're still healthy now. Maybe probably they are, because the chances are not that high that huge damage was made. And official version was that he wanted to make it protective against HIV. Actually, I personally think he is a bit smarter. He had also another intention. Regarding the mistakes that he made, scientists probably would say it is like using wild type Cas9 that could have off targets, and also not only like it can miss and cut the wrong gene because the guide can partially match DNA in another place. Also by itself, if you actually like in the institute, I know some people experimenting putting Cas9 without any guides, and if it doesn't have any guides at all, it very rarely, but sometimes actually cuts just for fun, and it's not good. And for this reason, people created high fidelity Cas9 and also enhanced Cas9, and 3 years they were already available, and he should have actually used them. But the main mistake was that he got funding from wrong guys who were not friends of Xi Jinping, and when they spoiled the relationship, he made him go to jail. And there were other issues. So when you do stuff, you also can get the organism mosaic, like not all the chromosomes and all the cells got the edit. And the other thing is that actually he didn't save them from a serious illness. Maybe if it was a serious disease that he would fix with Indel, maybe society would be more nice to him. And also, like any protein, CCR5 has both pros and cons. So it is entry receptor for HIV, but also it is a bit protective for some diseases. So first, what you do is you typically look at gene cards. That, like, if you have a gene, there are many sources, like you can look Wikipedia, you can look G cards. So if you want to edit a gene, you should check attentively what is there. And the second thing, what you really need is you also check for existing SNPs of the genes, if there are any associations. So for the benefit of this tutorial, let's try to actually reproduce CCR5 edit. And actually, when you Google the gene, you actually realize that, well, it's not that good in terms of HIV protection. It doesn't give 100% protection, but actually there is something that he preferred not to mention, but everybody knew, is that CCR5 is also a gene for some reason associated with enhanced learning, low hepatotoxicity, and is a bit protective against atherosclerosis. If you go so there is a database made by George Lab where he collects so-called superhuman gene, the genes that are super good for something. And he has like a blog that he found on human studies, and also he checks if those genes were really good for something in animals. And he also had specific part for cognitive related gene therapies, because actually that's what we humans love to do, to make our children and ourselves smarter. And in case of this gene, you can see that CCR5, on animal models, it was had enhanced learning, but it's not only on animals. There were studies on humans that actually proved that it's also good for cortical plasticity on humans. And the major problem, I believe, is that because of one bad old story with one German guy, things about human enhancement are sometimes treated badly by public media. And that's why instead of just publicly telling, I want to make kids smarter, he had to focus on another good property of the gene about HIV protection. And so let's make first the ugly interventions. So this gene, actually, he just broke it. And what I suggest is, so there are like 50 different tools to do it, because it's very simple to, because it's very simple to do the edit. Many people implemented the tools. And where is the chat? So please, let's just check, for example, Syntego, but you can check any other online tool. I give this link. If I will find where my chat is hidden, I will also copy paste it there. And let me actually do it as well. So first, what you do, you select Homo sapiens as a genome, then you do CCR5, and you do search. And so usually for CRISPR, you need to have, if it is Cas9, it has, it should be like 20 nucleotides long guide RNA. And before it, you should have a motif. So it should be like NGG. So any letter, NGG. And that's why so here they showed by default recommended guides, but we can actually see all the guides. So if you look into the guide, actually I think it's 19 nucleotides. And before, so we have these 19 nucleotides. And in the genome, before these nucleotides, you have a motif like any nucleotide and GG. And that's why, and for the scores, they give scores, usually it's several popular algorithms like Doench algorithm, like Azimuth. I think Syntego uses Azimuth. It is a model trained with Microsoft. I think they had something like random forests, and it looks into the sequence and flanking sequences. So 19 nucleotide guide and 10 nucleotide sequence on the left and right. And here you get the scores. So you see how should it cut on target score, and off targets, the format is like 0 direct off targets, 0 off targets with 1 nucleotide difference, and 2 off targets with 3. And it suggested you to select 3 best guides. And also they looked into genes. So in genes you have different regions, like promoter, it's like start. Then if somebody is asking, please do in written form. And
c'est pas de l'eau, c'est plutôt
mute French people. I don't understand French, unfortunately. And so far, so it's coding region, the regions that will turn into proteins. It's in exons, and that's why we focus on destroying the exon. And usually with indel, we want to make a frame shift or to make a stop codon. And so here we have the guides, and we actually have to put it in a viral vector. And what about the delivery? So there are different ways. If you do on cells, easiest is electroporation, and there are very cheap designs for in vivo electroporators. But as Ukrainian, at least according to Russian propaganda, I should be good with bioweapons. I would prefer to put it in a viral vector. And to put it in a viral vector, you have to use a service. So I will put a link to the chat. So I personally use Vector Builder because they have nice cloning and viral vectors production services. So in terms of viruses, there are many of them. The most popular ones, let me make it larger, and also can also open it yourself with the link, are AIV, It's adeno-associated virus. It's the safest one, but actually it's of very low efficiency. We tested them on cells in the lab, and we did optimization, like selecting the best serotype. So the serotype is actually those spikes that interact in fibers that interact with the cell and provides the entry. And from our experience, adenovirus was much better, and it doesn't integrate into genome. So it delivers as RNA and transcribes and translates the protein of your choice. People also, quite popular choice is lentivirus, but lentivirus integrates into DNA. And if we want to reproduce what's like gene editing, say on humans, we probably don't want additional integration of some lentiviral part. And they actually have vectors for CRISPR delivery. So that's why I'm actually checking mammalian CRISPR gene editing vector. And what we need to deliver both Cas9 that will do the cut and guide RNA. So I select adenoviral. So starting price is $269. And they have also really useful guide to show how it works, what are the pros and cons. So here I will just go to design. And here I have a choice. So actually from the research, it's recommended to use 2 or 3 guide RNAs to for sure knock the gene down. So I select dual gRNA vector. And here I have the design. So what you see here is a plasmid map, and it's a circular DNA. And this is used to produce, so you put it to some cells, like it can be, usually people use HAC cells to produce adenovirus. And what is important for us, we don't really care about most of this part. It's like technical, like origin replication, but we have regions that will be transcribed to RNA. So if we want to get RNA expressed from the plasmid, we use U6 promoter. So promoter just tells that please read something after me into RNA usually. And let's return to where we selected the guides. I believe I had this window somewhere because we have to copy them. Maybe it's better for me to close redundant papers. So I had the, yeah. So let's select best guides. This one looks really good. And the first one will be probably, wow, it's Zoom thing is really invasive. Okay. Okay, so the first one probably be the highest score. So I, hello world RNA. And for the sequence, we actually, it already did for us because it was in RNA format. In RNA format, you have uracil instead of thymine. And when I copy pasted, the vector builder actually figured out it and made the substitution. And what it gives to me, so here is my guide RNA. And also to make CRISPR work, I have a technical scaffold that is required to attach it to the protein. Luckily for me, in the plasmid, they already have it inserted, and that means that the only thing that I have to do is saying I'm happy with this design, add it to vector, and then I need the second one. I can also I can copy any of this. Usually, like if you have on target more than 0.5, it's usually reasonably good. And I let me like insert it. No, it's will be like second guide. And I'm happy for the vector. And I need to so they have like official name of this. So they already use this guide probably. Okay, so I have to select proper CRISPR, and I actually, so what they suggest is, so H means human, but it's just Cas9 that they codon optimized for human, just not really important. And they also added some text for purification that we don't really care about, and nuclear localization signals that just helps it to get into the nucleus. So two other options is, so here you have again CRISPR, but you have these two things. So in plasmid construction, you usually separate with, usually actually you do it with dash, not with colon, but here they did it with colon. So you have three elements here. Human Cas9, then you have P2A. P2A is just self-cleaving peptide. It means that if you want to make a trick to express several proteins from the same gene in mammalian cells, you cannot do just, you need, usually it's one promoter and one protein expressed, but if you put this P2A and then put second protein, it will express all this first as one protein, and then will cut them here into two. And the third part is just glowing. That means that it gives CRISPR and it gives glowing. And if we want to do some debugging, like to see if our child gets most of the cell got CRISPR, we would probably prefer to also have some glowing for debugging purposes. Although it's not really recommended. It's better people debug on cells, and when they go in vivo, they go without glowing, because it can a little bit damaging. So we got our vector, we push finish design, and we finally get, why is it taking so long? Okay, so nice. We got it. It will be produced, so it will take 20 days for them to prepare and $369. But we have problems that it's only like the DNA that they are making for us. And we have to actually add the packaging into viral particles. So we have to add this downstream service. And it means it will cost us $600 more approximately. And it takes, I think, around 20 extra days from 20 to 40 extra days to make those viral particles. So we will be able to just inject them to wherever we want. And that's if we that's how things work for CRISPR viral delivery. And coming back, this panel is so annoying.
So, and I'm sorry for interrupting you, but we don't have more time. Please try to.
Okay, so yeah, so we finished Hello World, and then what is important actually here is that I mean, I showed you not the most optimal way, because AD5 is somewhat immunogenic and you have to actually use a bit modified viruses in vivo. And overall, you should wait maybe 5-10 years when it will be useful and safe on humans. And also some people, it's also important, don't try it on yourself until at least you will check that you don't have immunity, because Streptococcus is immunogenic and its Cas9 can, in 20% people, can cause immune reaction. And actually, it's the ugly way of doing it, because the technology is pretty bad. And what I would suggest doing, so if we just want this receptor not to be expressed, we actually don't really need to kill it and to alter our genome, because we can do, we can use epigenetic CRISPR. So what people did, they mutated, they created dead Cas9, so they broke cutting ability, so it can only sit to DNA and do nothing. And the second thing is that they added modifiers to this protein that can, in this place of DNA, do some job. It can be activation, suppression, or if you put methylation, it will memorize this methylation, and this gene will not be active, will not be expressed for more than 50 days at least. And it's the way minimum people didn't test years. But if we want to do it with epigenetic CRISPR, it's pretty much similar. The major difference, I don't have time to go through these details, but it should be near start of transcription. Start when you, after promoter, you start transcribing DNA to RNA. If you put it somewhere close, plus minus 500 nucleotides, it will silence this gene and it will be heritable, and it can be cancelable by another epigenetic CRISPR or other mechanism if you want. And I don't have time, but there is CRISPR Era and other services to, you just use services for inhibiting CRISPR. And then, because it's not on vector builder, you go to add gene and you have to get this plasmid and send it to vector builder for further cloning, because vector builder, or you have to synthesize customly because they don't have template for epigenetic CRISPR. And the last thing is the proper editing, and the proper editing is actually CRISPR prime editing. Again, it's, I don't have time to go into details, but the idea is that you use nicases that cut only one strand of DNA, and you use a very tricky PEG RNA. That means that, so they've used that Cas9 with reverse transcription, that you give RNA to go to appropriate place, and you also give part of RNA that will be immediately turned into DNA. And because of very partial one-stranded mismatch repair, that is not really dangerous in comparison with double-stranded DNA repair, it will allow incorporating the sequence you want, and it's a bit less efficient, but it is much more safer than regular CRISPR HDR editing. And people even investigated and the stuff and improved it with combination with integrases. You can get inserted part with integrase site, and integrase a protein for actually originally also used in integrating phage DNA into bacteria, and it was repurposed, and you can get like 20,000 nucleotides inserted wherever you want. That is pretty cool. I give links to the papers. And this is comparison with HDR. So you can see that indels, the kind of dangerous guys, So it has so HDR is a bit more efficient than paste, but number of indels is crazy. So it's better to do paste multiple time than to use regular HDR. And that's the takeaway with CRISPR. And now you have learned how also to edit the genome. And thank you for your attention and happy for your questions.
I think there are questions in chat, and I don't know how much time we have left, but if you may answer them, that would be awesome.
Yeah, Anton, we have maximum 5-10 minutes.
Yeah, so so the service, I think you have to like, I mean, when you deliver in dry ice, actually, like every person cannot get like account and dry ice delivery. Usually they check who you are. So you can quite easily get design and get quote, but like if you want to deliver home, you can have issues with delivery, and delivery can cost a lot. Like dry ice delivery can cost like $1,000 easily. It's a crazy the way how prices are made in dry ice delivery. For Liz Parrish paper, yeah, I saw it. They use another type of virus. It was, I think it was leukemia M something virus. They used the delivery through ice. So actually, it's quite common to deliver viruses through nose or ice. I don't remember. I think it was nose actually, nasal injections. Yes, so and they actually had H-third, and they had a second gene that is follistatin or something. My memory is pretty bad. For those people who did CRISPR on themselves, so the question how efficient it was, actually it's not that super easy to deliver it efficiently. The easiest thing is if you want just to deliver to skin, like for example, you want to make glowing skin, like you deliver GFP, you do in vivo electroporation. It's quite painful. There are open source designs that you can just within $100 for consumables actually assemble this electroporator, but it will first damage your skin. Second, you have to check if you don't have autoimmunity against Cas9, otherwise you will be in a big trouble with adverse reaction. And third, I would actually recommend always to try on cells or animals, because then on yourself. What else? I know, I think it's like all questions. I don't see any other questions.
Wait, what do you mean try somewhere? You received this CRISPR. Can you multiply it to use?
The thing is that there are different ways of delivery. So I don't recommend to use viruses, at least the method I showed to you. Like adenovirus is highly immunogenic. Even if you use gutless adenovirus, better don't try it on yourself. So if you get viral particles, you can inject them, but they have mutations that don't allow to multiply them. So you need helper plasmid to multiply. So you cannot, you get some number of particles, but you cannot multiply them unless you will put helper plasmid, and you'll have a cell line that will be producing. For things like injecting, like electroporations that you can do probably with skin, there you actually don't need a virus. You just need to buy, it's pretty easy to get some plasmid DNA, but efficiency, I don't know. I mean, you will probably reach only to part of the skin cells. It's a common problem in vivo, and you will hurt your skin because of electricity. And I don't have expertise with in vivo electroporation to say how many cells will get CRISPR. And it's of a limited use. I mean, okay, you modify part of your skin that you also damage with electricity. So what stuff you get? What's useful stuff you can get at this small patch of skin?
That was the question. Are there any useful use cases?
Well, I mean, you probably can deliver elastin. So if you, because usually it goes down with age, but maybe you can deliver some regenerative things. But the problem also is that it will not be really specific. And if you have different cells, like for example, elastin can be good for skin and arteries, but I don't think like neurons will benefit much from elastin. So I mean, things are complex, and even though it's not that super hard to do something stupid, in most of the cases, it will just not work.
So the most people who order this on this paid services, they just do some experiments.
Yeah, most of them are, because you can spend a lot of money to make some fun, but usually you kind of have to plan really hard and experiment a lot to get results. So maybe you can do something on your kitchen, but the easiest thing is just to make things glowing. But okay, you made it glowing, but what is the applicability of it? Go to a party and show? Maybe that's it. And for regeneration, it's hard. You have to do a lot of research. Like you can deliver the factors, but you have to understand where, how. And so the stuff I gave is just for fun. I thought maybe. Okay. Okay, guys, so I see no more questions. So so far, every of you will, question. Okay, Arseny, you will be the last one. Larissa needs to sleep, Okay, Arseny, last question. So Vector Builder is in China, actually. Actually, in Europe, it's a limited number of companies that you can deal with. But in Vector Builder, things go from China. And it's actually a big headache because of transportation. In European Union, there is a viral core in Barcelona, but you have to wait 6 months until they will do anything. Really annoying. Okay, guys, I finished, so maybe we can close it.
So I would like to thank our speakers. Thanks, Anton, and thanks your team and Kim. And also, I'd like to thank our participants, and I'm looking forward to see you again to our next webinars.
Very well.
Bye.
Thank you as well.
Bye.
Bye.
Pleasure.
See you.
Thank you.
Nice evening.